जैसा कि आप जानते हैं हमारे पेपर वन के रिगार्डिंग क्रैश कोर्स चल रहा है एंड इन दिस वीडियो वी विल डिस्कस कंप्लीट बायोलॉजी। बायोलॉजी के हमारे एग्ज़ाम में इंपॉर्टेंस है कि 9 से 10 क्वेश्चन डायरेक्ट देखने को मिलते हैं एंड साइंस इज़ एन इम्पोर्टेंट सब्जेक्ट, हम इसको नेगलेक्ट नहीं कर सकते हैं। स्कोरिंग सब्जेक्ट भी है। लास्ट 2 से 3 सालों से साइंस काफ़ी ईजी टू मॉडरेट कैटेगरी में आ रहा है एंड वी हैव टू टेक द बेनिफिट ऑफ दिस सब्जेक्ट ऑल्सो। इसके रिगार्डिंग ऑलरेडी पेपर 2 की फ्री सीरीज भी चल रही है और टेलीग्राम चैनल पे हम रेगुलर बेसिस पे पेपर 2 के टॉपिक्स पे डिस्कस करते हैं। ऑल्सो द इम्पोर्टेंट इन्फॉर्मेशन कि 12th मई से हमारा पेपर 2 का डेडिकेटेड बैच भी स्टार्ट होने जा रहा है। दिल्ली में ऑफलाइन ये बैच रहेगा, ऑनलाइन मोड में भी अवेलेबल है। इफ़ यू वांट टू प्रैक्टिस द टेस्ट सीरीज तो प्रतिभा टेस्ट सीरीज इज देयर। इट्स ए comprehensive टेस्ट सीरीज फॉर योर पेपर 1 एंड पेपर 2। इट ऑल्सो इंक्लूड्स योर मेंटरशिप पार्ट, आफ्टर एग्जाम गाइडेंस भी इसमें इंक्लूड है। इट इंक्लूड्स 55 टेस्ट, जिसमें 35 टेस्ट पेपर 1 के रहेंगे, 20 टेस्ट पेपर 2 के रहेंगे। सारे टेस्ट आपके इवेलुएट किए जाएंगे। आप इवेलुएटर से बात कर सकते हैं। किसी भी और तरीके की गाइडेंस आपको चाहिए आप कमेंट सेक्शन में बता सकते हैं। वीडियो कंप्लीट होने के बाद कंप्लीटेड लिखना मत भूलिएगा और नेक्स्ट किस सब्जेक्ट पे आपको वीडियो चाहिए तो कमेंट सेक्शन में ज़रूर बताइएगा। तो मिलते हैं नेक्स्ट वीडियो में। जय हिंद।
The basic concept in biology is about cell. All organisms are made up of cells. So cell is basically the fundamental unit of life. Cell is the fundamental unit of life. Now, whether you take a whale or a microbe like bacteria is made up of one cell. Elephant can have billions of cells, human can have billions of cells. But at the end, in each organism, it is finally made up of units called cells. So this is a Latin word, means a little room. The cell was, or this a room-like structure was observed by Robert Hooke when he was taking a look into the cork cell. He observed those spaces and he called them cell. And he called those spaces as the cell. Then later came cell theory. This was given by two fellows, Sleeden and Swan. Two fellows gave the cell theory called Sleeden and Swan. So basically, it says that cell is the basic unit of life and cells emerge from pre-existing cell. There were a lot of dispute or lot of discussion regarding origin of the cell. Okay, there were a lot of discussion regarding origin of the cell. Some theory said that life emerges from a decaying material. So if decay, they said that maggots are formed when the decay is happening. So several experiments were carried out and this disapproved all those things. So basically cell emerged from pre-existing cell. So cell is the basic unit of life. All the large animal would be made up of billions of cells. So cell is the fundamental unit. Second, cells emerge from pre-existing cell. Now, how we study cell? For studying cell and its component, we must have the capability to take a look at the finer structure. So for that, we have what? We have microscope. So microscope can take a look at the finer structure. You have this eyepiece. Another is the objective lens. So they magnify the specimen is kept on the slide over here and they magnify and then you say a larger image. That is how cell, its component and other thing was being studied. Then as time moved ahead, we have advancement and after light microscope, we have emergence of a thing called electron microscope. So in electron microscope, we are using electron beam in place of light. Electron microscope use electron beam in place of light. So basically these electron beam strike the specimen, then they are deflected and then we record things. So it can see things at very nano scale, 10 to the power -9 meter. Now we have very small structures like ribosome. Ribosome has a size of 20 to 25 nanometer. So using these electron microscope, you can visualize even very small particles. You can visualize small particles, that is small cells, even the cell organelle and other components inside the cell. Now, the shape and size of the cell depends on the function. It is not fixed. Inside our body, several functions go. Eye has a different function, ear has a different function, kidney has a different function, liver has a different function. So the shape and size of the cell depend upon the type of the function it has to perform. For example, take the case of nerve cell. The function of nerve cell is to transmit signal. So it is elongated like a wire. Since its function is to carry signal from one end, say from my hand to my brain. So it must be like a wire-like structure. So it is elongated. Muscle cells are also elongated. Whereas a red blood cell is circular. Why? Because the blood cells have to move through the blood capillaries. They have to move through the blood capillaries. And that is why they are circular in shape, so that they can pass through the blood capillaries, arteries, veins, etc. Again, take a look over here. Egg cell are round, they are larger, they do not have a tail because they are stationary, whereas the sperm cell have a tail. Why? Because the sperm cell move inside the reproductive tract in the female, and then they fertilize the egg, and then zygote is formed. So the shape and size of the cell depend on function. Largest egg is an ostrich egg and the smallest is of mycoplasma. Even within the cell, you have different component performing different function. In any organization, you have different people performing different role. A big organization, finance department is different, HR is different, marketing is different. Similarly, inside the cell, you have different department or different component or organelle. They perform different function. Okay. So these are called cell organelles. In our body, we have system, and in system, we have organs. So comparative to that, inside the cell is what? Organelle. So each organelle performs a specific function, and that is why a cell is able to survive or perform all its duties, because they work in unison. Now, I am giving here this is an animal cell. This is a diagram of your NCERT animal cell. We have two cell, one is animal, another is plant cell. Basically, at present, I am limiting myself to animal and plant cell. So this is an animal cell. Just take a look over here. Here you have the nucleus. Outside the nucleus, you have your endoplasmic reticulum. These are tubule-like structure attached to the nucleus. Just near to the nucleus, you have these tube-like structures. And if ribosome is attached, it is called rough endoplasmic reticulum, and if ribosome is not attached, it is called smooth endoplasmic reticulum. Then you have Golgi body. Then you have lysosome. Outermost layer is plasma membrane. Okay. Then you have mitochondria, etc. Now compare it with an animal cell. In animal cell, outside the plasma membrane, you have a structure called cell wall. So cell wall is outside the plasma membrane. One major difference is there. Here another difference you will see is the size of the vacuole. Sometimes the size of vacuole is very large. Here you have Golgi, mitochondria. Plant cell have a unique thing, that is chloroplast. Plant cell can make their own food. That is why they are autotroph. They can make their own food. Animal cell cannot make their food. That is why animals are dependent on other. Whereas in case of plant have the capacity to make their own food. That is why they are autotroph. They can make their own food. Now coming to the first organelle in the cell is plasma membrane. The outermost layer of the cell is what? Plasma membrane. So outermost layer of the cell, plasma membrane. Some of the key point is, see, the internal environment of the cell will be different from its external environment. So how this is done? This is done through your plasma membrane. It maintains a differential between inside the cell and outside the cell. So inside of the cell, if everything is same, then there would be no difference between inside the cell and outside the cell. So for maintaining a difference between inside and outside, the cell membrane must allow few things to come in and restrict other, say A, B, C is allowed, but D, E, F is not allowed. So this is called selectively permeable or semi-permeable. Selectively permeable. You are allowing something to come inside, you are blocking something others. So that is how you maintain a differential between inside and outside. Now plasma membrane is made up of lipid and protein. Sometimes you have the term lipoprotein. Then on the surface of plasma membrane you have receptors for hormone. Many of the hormones do not directly go inside the cell but get attached to their receptors. For example, here it is written receptor protein. So for many biomolecule, even for viruses, even for drugs, receptors are found on the plasma membrane. So many of the drugs can also interact, many of the biomolecules, many of the hormones can interact in this way. So they get attached to the surface, and these are called receptors. Receptor for hormone and biomolecules are present on its surface. This is the biological or chemical constituent of the plasma membrane, that is lipid and protein. Or sometimes we have a word called lipoprotein. Sometimes we have a word called lipoprotein. Take a look over here. You have a hormone called insulin. So insulin receptors are found on the plasma membrane. So insulin hormone, impact of insulin hormone will be visible only when the insulin hormone gets attached to the receptor. So for many drugs also you have receptor protein. Many drugs also go at in different organs you have cells, say intestine, other places. So many of the drugs also go and attach with these. Then we have cell wall. Cell wall is not found in animal cell. Cell wall is found where? It is found in plant cell, bacteria and fungus. Now here I am focusing on plant cell. So this is found on the outer side. On the outer side, cell wall is made up of primarily of cellulose, hemicellulose and pectin. And cellulose is a complex substance. That is why it gives animal cells have greater strength, or they have greater capability to survive in an environment. Why? Because they have presence of cellulose. So they can withstand greater variability as compared to the animal cell. Now just take a look as over here. You have cellulose, hemicellulose and some other things over here. Okay. Now in the center of the cell you had nucleus. Whether it is a plant cell or on the inner side you will find this is the animal cell. So animal cell have cell wall and this is vacuole. Sometimes they displace the cytoplasm to one side, and here you have the nucleus. So after cell wall, coming to the nucleus. In nucleus we always think about chromosomes. That is not wrong. We always think about what? We always think about chromosomes. Okay, chromosome would be there. But the nucleus is the control and command center of the cell. Here you have nuclear membrane, and gap in the nuclear membrane is called nuclear pore. So through nuclear pore, things can go from cytoplasm to nucleus, and things can come out from nucleus to the cytoplasm. Okay. Now you have thread-like structure over here. It is like a piece of thread that is open, and you have a structure called nucleolus. Nucleolus is rich in RNA. Nucleolus is rich in RNA. Now, when the cell is not dividing, the DNA is present in the form of these thread-like structure called chromatin. In a non-dividing cell, you will not see chromosome. In a non-dividing cell, you will not see chromosomes. The DNA would be present in thread-like structure, and this structure is called chromatin. only when the cell is about to divide, these chromatin get arranged into the chromosome-like structure. Okay. So these chromosome become visible only at the time of cell division. Otherwise, the DNA is present in the form of thread-like structure, and this structure is called chromatin. This structure is called what? This is called chromatin. So at the time of cell division, the chromatin condense and they form the specific structure that is the chromosomes. Now chromosomes have two arms. It is attached at the center called centromere place. So chromosomes carry your hereditary character. We have 46 chromosome, 23 chromosome coming from father, 23 chromosome coming from mother. But when the cell is not dividing, this will not be visible. This will be in the form of what? Thread-like structure called chromatin. Now, long back, this chromosome is made up of two things, DNA and protein. The chromosomes in the organism are made up of two things, DNA plus histone protein. Remember, one unique protein is there in DNA called histone. Histone protein is there in case of DNA. Now, in your school days, if you remember certain diagram from your school days, you have this structure called double helical structure of DNA. Two helix. DNA is like two helix going up. This structure of double helical structure of DNA was given by Watson and Crick, for which they won the Nobel Prize. So, in your 10th and 11th book, you have reference of this Watson and Crick model of DNA, James Watson and Francis Crick. So, James Watson and Watson and Crick got the Nobel Prize for giving the structure of your DNA. Now, moving into the chromosome. So, we discussed chromosome are made up of two things, DNA and protein, particularly histone protein. Now, the DNA is the blueprint of the life. DNA is what? Blueprint of life. They decide your height, color, weight, and other body function. So, they carry the information so they are in the form of coded information. That is how your body is controlled. Now, on these chromosomes, you have functional segment. The chromosome control your character. These are in the form of genes. So 100% of the DNA is not making any particular protein. Segments of these DNA produce a particular protein, and these functional segment of DNA are called genes. So genes are the basic unit of heredity, and they carry instruction for protein synthesis. They carry instruction for protein synthesis. So they are arranged linearly on the chromosomes. It may not be, there can be gap between two genes. The nucleotide or the DNA is there. So DNA is continuous, but at one place it is making protein, then there is a gap, again it is making protein. So there can be like that. Chromatin we discussed, that thread-like structure. Then during your cell division, they get arranged into chromosome. The ends of chromosome are called telomere. The two arm of the chromosome. So here, when the cell is about to divide, DNA makes a copy of itself. There are two copies of DNA here at the time of cell division. And the two copies are attached at this place called centromere. The ends of the chromosome are called telomeres. They have their own function. Telomeres like as a protective cap. Now, in case of bacteria or prokaryotes, the nuclear region is not clearly defined. Why? In some organisms, a distinct nuclear membrane is not present. In some organisms, a distinct nuclear membrane is not present. The distinct nuclear membrane is not present. So, what happens when the distinct nuclear membrane is not present? the nuclear region is not clearly defined. The nucleic acid is slightly dispersed in that area, and such structure is called nucleoid area, nucleoid. So, undefined nuclear region is called nucleoid, nucleoid, and such animals are called prokaryotic. See, in many of the books it is given nucleus is absent, but what NCERT writes is, and yeah, the nuclear region is not clearly defined. It is not saying that nucleus is absent, but it is writing that it is not clearly defined nuclear region. Okay. So those animal which have nucleoid, that is the nucleic acid or the genetic material is not entirely covered by a membrane, they are called prokaryote. And those organisms where a distinct nuclear membrane is present is called eukaryote. So as life evolved, from simple to complex things came into picture. So in the beginning, nucleus was not there. Later, what happened? The nucleus comes into picture. Okay, the nucleus comes into picture. That is there. Now, many of these prokaryotes also lack many cell organelles. We discussed that just like organs in our body, inside the cell we have component which perform a specific function. These are called organelles. These are called what? Organelles. So prokaryotic cell lack many of the organelles also. Now moving on to the chromosome number. The number of chromosome is fixed in each organism. For example, in humans, we have 46 chromosome. 46 chromosome or 23 pairs. We say 46 chromosome or 23 pairs. We have 23 pairs of chromosome. Okay. Now this paired condition is called diploid. They exist in pairs. In human, male produces sperm, female produces ova. In sperm and ova, the number of chromosome is not 46, but it is half, that is 23. They have one pair or one set of chromosome, and this condition is called haploid. So the gametes, that is sperm and ova, have haploid condition of one set of chromosome. Two set of chromosome or diploid are found where? In case of normal cell. In some of the plant, you have multiple sets of chromosome. It could be 3 pair, 4 pair, 3 set, 4 set. This is called triploid, tetraploid, etc. Then moving on to the genetic material, there are two kind of genetic material. One is RNA, another is DNA. So let us first come to RNA. RNA has ribose sugar, DNA has deoxyribose sugar. RNA has what? Ribose sugar. Then these sugar molecules or these sugar, we know about a general sugar called glucose. Very common, glucose is an example of sugar. Here we have another kind of sugar. This is 5-carbon sugar called ribose. Second is your bases containing nitrogen. Nitrogenous bases, we have 4 kind of nitrogenous bases. AUGC, adenine, uracil, cytosine and guanine, AUGC. So in RNA we have 4 nitrogenous bases: AU, adenine, uracil, cytosine and guanine. So this is mostly single stranded. DNA is double stranded, but RNA is single stranded. Okay. In general, but RNA can also be double stranded and can have complex structure. See, in evolution, RNA came first. RNA was less stable. That is why RNA is the genetic material only in some of the viruses. In right from bacteria, from prokaryote to the higher animal, the genetic material is what? DNA. So only in some of the virus. So virus can be either RNA virus or DNA virus in higher animal, and RNA is less stable. So in fact, during the period of COVID is an example of an RNA virus. Now, if you remember, at that time we had a vaccine called Pfizer and Moderna vaccine. There were different kind of vaccine, and these were basically mRNA vaccine. Since RNA is unstable, they need to be stored at very low temperature. That was one of the challenge. Now there are three type of RNA. One is messenger RNA, second is ribosomal RNA, and third is transfer RNA, mRNA, rRNA, and tRNA. There are three type of RNA: messenger RNA, ribosomal RNA, and transfer RNA, mRNA, rRNA, and tRNA. So basically, from the name itself, messenger means what? It is carrying message. So basically, it carries the message from the DNA for protein synthesis. So on mRNA is formed on DNA. So mRNA carries the message for protein synthesis. mRNA carries the message for protein synthesis. Protein is nothing but a chain of amino acid, one amino acid attached after the other. So basically on DNA, you have formation of mRNA. On DNA, you have formation of messenger RNA. This is the DNA. Here mRNA is being formed, and based on the messenger RNA message, protein is synthesized. Protein is nothing but a chain of amino acid. So protein is a chain of amino. There is a great Indian scientist, Dr. Hargobind Khurana Sahab. He got one of the Nobel laureate. He got Nobel Prize long back in the 60s, I think 70s or something. I don't remember the year. So he deciphered a thing called genetic code. So genetic code is basically correlation between three nucleotide on mRNA, correlation between three nucleotide on mRNA, messenger RNA, three nucleotide on the messenger RNA, and the amino acid that is added. I will just show you. For example, take a look over here. The first three letter in the messenger RNA AUG. Now this AUG code for methionine, MET. Then the second three letter is CCG, so second amino acid. Then UAU, this is the third triplet, means three nucleotide. This codes for this one. Then the fourth, like this, total 5 amino acid are coded over here. It could be longer also. So basically, the genetic code is finally converted into the protein thing. The genetic code is finally converted into what? Protein. So based on the three letter code, amino acids are added one after the other. Then you have transfer RNA. So the function of transfer RNA is to catch hold of the amino acid and carry them to the ribosome. Then you have ribosomal RNA, rRNA. Ribosomal RNA are found in the ribosome, that is the protein factory. That is the protein factory. Now take a look at the structure. This is the nucleus. Inside the nucleus you have DNA. On DNA formation you have formation of mRNA. We studied about DNA. This is the structure of DNA. Here you have the sugar molecule. Just take a look, 1, 2, 3, 4, 5. This is the sugar molecule or deoxyribose sugar. Sugar molecule are found on the outer side. Sugar and phosphate, PO4, and nitrogenous bases are found on the inner side, A, T, G, C. So in DNA you have 4 nucleotide: adenine, thymine, cytosine, guanine. They contain nitrogen. That is why they are called nitrogenous bases. In DNA you have deoxyribose sugar. In RNA you have ribose sugar. Then you have 4 nucleotide in DNA: A, T, G, C, adenine, thymine, cytosine, guanine. In RNA, thymine, in place of thymine, you have what? Uracil. Sugar here is There it is deoxyribose sugar. In case of RNA, it is ribose sugar. DNA is double helical most of the cases. RNA is mostly single helical, single strand, but it can be double stranded or it can have complex structure. So that is how life is regulated. On DNA, you have formation of messenger RNA. Based on the RNA, you have formation of protein. This is called central dogma of life. That is how life runs. That is how life runs. Now moving on to cell organelle. Moving on to the cell organelle, there is a structure called endomembranous system. Endo means inside, and membranous means it is made up of membranes. So you have three important structure: endoplasmic reticulum, ER, Golgi, and lysosome and vacuole. They have an interrelationship or they are connected with each other. That is why they are part of endomembranous system. Whereas mitochondria and chloroplast, they do not directly coordinate with them. That is why they are slightly different. They are also organelle, but they are not part of the endomembranous system. Okay, that we do not include them into. They are organelles, but they are not part of the endomembranous system organelle. They are organelle. Okay. The function of mitochondria and chloroplast peroxisome are not coordinated with the above component, and therefore they are not considered as part of the endomembranous system. They are organelle, but not part of the endomembranous system. Just take a look. This is the nucleus. You have formation of mRNA. It comes out, it gets attached to the ER, endoplasmic reticulum. So protein synthesis occurs over rough ER. Then a small vesicle like this carry both from rough ER and smooth ER, they carry it to Golgi. Certain changes take place in Golgi. Then either they can be thrown out of the cell or they can be transferred from Golgi to lysosome, and then lysosome will perform its function. So ER, Golgi, either to lysosome or they can be thrown out of the cell. This is called secretory vesicle or excretory vesicle. For example, in many of your glands, for example, salivary gland, there is continuous secretion of the saliva. There is continuous secretion of the saliva. So you have this structure. This is the ER, rough ER, and if ribosome is not attached, that is called smooth ER. Then you have Golgi. This is the Golgi body. Then from Golgi, it can either go to lysosome or it can go out of the cell as secretory vesicle. Okay. So ER can be of endoplasmic reticulum is attached near to the nucleus. They are made up of vesicles, seeds, tube-like structure. If ribosome is attached, that is called rough ER. If ribosome is not attached, that is called smooth ER. So rough Y, they are called rough ER because presence of ribosome. It looks rough on the surface. So in all the active cell, you have them. These rough ER are the place where protein synthesis take place. Rough endoplasmic reticulum have ribosome. The DNA, on DNA you have formation of mRNA. mRNA comes here, attached it is, and then ribosome makes the protein, and the protein goes inside the ER. So rough ER are the site of what? Protein synthesis. Then you have one is rough one, another is the smooth ER. Smooth ER, they produce fat or lipids. They are the site of the formation of lipids or fats. So both protein formation occurring on rough ER, protein on rough endoplasmic reticulum, and fat on smooth ER. So together the protein and fat, that is lipid and protein together form lipoprotein. So the plasma membrane or the membrane are made up of what? Lipid and protein. That is why ER are involved in membrane formation or membrane biogenesis. Bio means biological system, genesis means formation. So ER produces both protein as well as fat. So protein and fat, lipid and fat can combine to form what? Lipoprotein. Membranes are basically lipoprotein. Another thing is the smooth endoplasmic reticulum. They play an important role in detoxification of drugs and chemicals. See, when you take any drug or chemical, they finally go to liver. So these chemicals have to be transformed into harmless form, and this process is called detoxification. The process of conversion of a chemical into another form is called detoxification. This takes place at the level of SER, smooth endoplasmic reticulum. So basically, ER makes the material and then it goes to different place inside the cell. Now coming to after ER, the things come to where? Golgi. So from ER, the things came to where? It came to Golgi body. From ER, it arrived where? It arrived at the Golgi body. The material synthesized near the ER is packaged and dispatched to various targets inside and outside the cell through Golgi body. So from ER, small vesicle will carry these product to where? These product to the Golgi apparatus. That is carried to the Golgi apparatus. Now in Golgi, what happens? They are stored. The product which are produced at ER are sent to Golgi. There they are stored, modified, and then packed, and then they will further be sold, sent here and there. So storage, modification, and packaging takes place where? In the Golgi. Now, say you have simple sugar, you can convert them into complex sugar, glycolipid, glycoprotein. You attach sugar to lipid, it becomes glycoliquid. You attach sugar to protein, it becomes what? Glycoprotein. So these things are done over here. Then, so that is the site of formation of glycoprotein. Then you have formation of lysosome. Basically, from ER, the lysosomes are formed. So they split off, the enzymes are formed here. So they dispatch and go and become the lysosome. So we started with ER, then we came to Golgi. So basically, it is the main place of sorting out, packaging, and then modifying and then sending to the cell. Just like a factory, the cell also has different workstation. So some work is done at first workstation, then second, then third. So work started at where? At ER, then it came to Golgi. From Golgi, it will go either to lysosome or it can go out of the cell. That is there. Then let us come to lysosome. So lysosome, you all know it is called suicidal bag of the cell. We'll come to that also. Now lysosome are membrane. They have outer membrane layer and it contains enzymes, hydrolytic enzymes. Hydrolytic. Hydro means water, lighting means breaking up. It will attach a water molecule and it will break the thing into two. So it can be protease, carbohydrates, like almost all component of the cell can be broken down by the enzyme of the your lysosomes. So basically, any component of cell that becomes old or non-functional or which is not performing its duty can be broken down and can be digested by the lysosome system. So that is why it is also called waste disposal system of the cell. Say a bacteria is taken up by a cell, the lysosome will come and try to digest the bacteria. Any foreign particle or any foreign organism entering inside the cell will be digested by it. But why? So that is, it has protective role. Now why it is called suicidal bag? See, sometime certain disturbance can take place in the cell. Certain disturbance can take place in the cell. So when the disturbance takes place inside the cell, what happens? That the membrane gets damaged and the enzyme comes out of the lysosome and it digests the cell itself. That is why the cell itself gets destroyed, and that is why it is called what? Suicidal bag. It is called suicidal bag. Then they play very important role in a thing called a phenomenon called metamorphosis. A phenomenon called metamorphosis. I will just come to metamorphosis after this slide. Take a look at this. This is the silkworm. Here it is the egg. Then comes caterpillar or larva. Then it is a voracious eater. Then it becomes pupa. It gets enclosed inside the silk threads. And after some time it cuts and then the butterfly comes out. So all four are the same organism, but they look quite different from one another. In lower animals, you have a phenomenon called metamorphosis, where stages of life are quite distinct from each other. One stage of life, for example, in case of frog, this is silkworm, take another case, frog. The young one of a frog is called tadpole. Tadpole has a tail. When it becomes adult, the tail disappears. The tail disappears. So, this process of metamorphosis occurs with the help of lysosome. So, that has been asked in exam. Remember, that has been asked in the exam, that lysosome plays a role in metamorphosis. Lysosome has protective role. It can digest microbe. Lysosome is also waste disposal system of the cell. Okay. So, UPSC has asked that. It is waste disposal system of the cell. So, there is change of stage. What? From egg, larva, pupa, imago or butterfly. You get silk from cocoon or the pupa stage. Okay. You have different varieties of silk, tassar, mulberry, etc. Eri, mugar. The scientific name of silkworm is Bombyx mori. This has been asked by in several exams by UPSC, remember. So basically silk you get from which stage? Cocoon or pupa. Second, scientific name of silkworm, question on that, Bombyx mori. Assam has a unique silk called Muga. This is found only in Assam. Then come to another very important organelle, that is mitochondria. This is called powerhouse of the cell. Powerhouse of the cell because it produces ATP, adenosine triphosphate. That is the energy currency of the cell. That is the energy currency of the cell. So mitochondria, if you take a look at, it has double membrane, outer membrane and inner membrane. In this diagram, it is more distinct. It has an outer membrane and an inner membrane. The inner membrane is thrown into infoldings, and these infoldings are called cristae. These infoldings are called cristae. And here you have certain structure found on the inner membrane called F0, F1 complex or oxysome. This is the place where ATP or adenosine triphosphate is formed. ATP is the energy currency of the cell. So ATP is formed where? ATP is formed by the F0, F1 complex. Then you will find DNA. Two organelles, only two organelles contain DNA. One is your mitochondria and second is chloroplast. Only two organelles contain DNA and that is why these two organelles are considered to be semi-autonomous. Mitochondria and chloroplast have their own DNA and they also have ribosome. So primarily, since they have their own DNA, they are called semi-autonomous organelle. They are called semi-autonomous organelle. And they are the site of aerobic respiration. Finally, glucose is oxidized to CO2 and you have production of ATP, adenosine triphosphate. This is the energy currency of the cell, ATP, adenosine triphosphate. So outer and inner, the inner membrane forms what? cristae we discussed. The inner membrane is into infolding called cristae. You have formation of ATP, energy currency of the cell. Then I told you their own DNA, and that is why both mitochondria and chloroplast are semi-autonomous. Second, one important point is 100% of the mitochondria is from maternal side. We call it maternal inheritance. We call maternal inheritance. Maternal inheritance. Why? In formation of zygote, sperm fuses with egg to form zygote. Sperm fuses with egg to form zygote. Now, the mitochondria of the sperm are found in the tail. So, when the sperm fuses with the egg, it forms zygote, but tail remains outside. Tail remains outside. That is why 100% of the mitochondria comes from where? Egg. So, sperm is not contributing any mitochondria to the zygote. That is why there is maternal inheritance. So, if mother's DNA has some deficiency, all her children, whether it is a boy child or a girl child, will inherit those deficiency. Mitochondrial DNA has certain very interesting application. Two techniques are there. One is DNA barcoding and second is universal primer technology. Both these technology utilize mitochondrial gene. So in gene also you have what? Sequence, there are 4 letter only: adenine, thymine, cytosine, guanine, ATGC. So both these technology are based on mitochondrial gene. They find out the sequence of the gene and then sequence of ATGC on the gene, nucleotide on the gene, and then they identify the species. So DNA barcoding and UPT. UPT has been developed in India by CCMB, Center for Cellular and Molecular Biology, Hyderabad. They are used for identification of species. Then you have plastids. Plastids are found in plant cell. Now, in case of there are 3 type of plastid: chloroplast, chromoplast, leucoplast. There are 3 kind of plastid. Plastids are present where? They are present in plant cell. Chloroplast, chromoplast and leucoplast. So, these plastid also have double membranous structure. Let us first come to chloroplast. The outer membrane is there, you have inner membrane. Outer membrane and inner membrane. Here you have ground substance called stroma. Stroma is the ground substance. And then you have coin-like structure kept over one another. Coin-like structure kept over one another. Individual coin is called thylakoid and these together are called granum. The stack of coin is called granum and the individual coin is called thylakoid. The photosynthetic pigment is found on these thylakoid. Photosynthesis is done by the green pigment chlorophyll, and chlorophyll is present where? They are present on these thylakoid. Chlorophyll and carotenoid pigment trap light energy essential for photosynthesis. Okay. So basically the thylakoid contain the green pigment, that is chlorophyll and carotenoid. So the light reaction of the photosynthesis occurs in case of your thylakoid. So we have the stroma, that is the ground substance, and the coin-like structure. So these are the thylakoids. And one more thing, chlorophyll contains magnesium ion. This has been asked by the different exams, right from higher to lower exam by UPSC and other places also. Okay. So chlorophyll contains what? Magnesium. Chlorophyll contains magnesium. Chloroplast is considered to be semi-autonomous like mitochondria because it has DNA and ribosome. Then you will find that the mango is green in the beginning, then it can become yellow or red also, even in case of papaya. So there are three type of plastid: chloroplast, chromoplast, and leucoplast. Chromoplast are colored plastid. Their main function is color. The function of color is two things: attract insect. So flowers are colored. So colored flower, insect pollinated flowers are colored. They attract the insect for pollination. Second, they also attract animals and insect, and which helps in dispersal of seed. When the fruit becomes ripe, it is colored. So what happens? A yellow colored fruit will appeal to the animal. The animal will eat it. It will carry it somewhere far away or some distance away from the plant. So the seeds will be dispersed. So this helps in the dispersal of seed. Then third kind of plastid is leucoplast. Here the main function is storage. So many of the grains and other store. So it can be storage can be for carbohydrate. For example, that is amyloplast. In case of potato, rice, wheat. Certain cases it can be for oil, where the storage is oil or fat, that is called elioplast, leucoplast. Second and third, it could store protein, aleuroplast. For example, in legumes, pulses, legumes have protein, high amount of protein, all the pulses. So there protein is also stored, that is it. Then coming to vacuoles. Vacuoles are sacs for storage of solid and liquid content. They could be sugar, they could be protein, they could be XYZ, anything which the plant produces or the which is generated in the animal during a particular process. Okay. So basically production or photosynthesis primarily done by plant. So that is why vacuoles are prominent in case of plant cell, and sometimes vacuoles can fill more than 50% of the entire space of the cell. So vacuoles are more prominent in the plant cell. Take a look at this is the plant cell. So bulk of the space here is occupied by what? Vacuoles. Bulk of the space is occupied by vacuole. Whereas in animal cell they are very small. In animal cell they are very small. Whereas in case of plant cell, because plant cell produces various type of product, many of the biochemical product of the plant also have medicinal uses. They have medicinal uses. So vacuoles are prominent in your plant cell, less prominent in animal cell. Okay. So even in case of amoeba, so amoeba takes certain food, digest it, then it can have food vacuole. Vacuole can also have role in excretion. Some of the waste product are stored in the vacuole and then they are thrown out of the cell. Then you have ribosomes. So ribosome, they are responsible for protein synthesis. They are the protein factory. Ribosomes are protein factory. So basically on DNA you have formation of mRNA and finally you have formation of protein. So on DNA you have formation of mRNA, this is called transcription, and finally you have formation of protein, this is called translation. So first step is called transcription, DNA to mRNA, and the second step is called translation of protein synthesis. So that is how life is regulated. On DNA you have formation of mRNA, the ribosome reads the mRNA, forms the protein. So the protein factory is what? The protein factory is your ribosome. So ribosome are made up of two things, RNA and protein. They are the protein factory. They are the site of protein synthesis. So the message on the mRNA, messenger RNA, is finally converted into the functional molecule, that is protein. Now ribosomes, in case of prokaryote and eukaryote, are variable. Prokaryote ribosome are 70S, eukaryote are 80S. 70S has two subunits, 50 and 30, and 80S has two subunits, 60 and 80. So eukaryote have 80S, prokaryote have 70S. Okay. So Nobel Prize Chemistry 2019 is for study of ribosome. Now, why I mentioned here, because you have a one Indian scientist, Venkatraman Ramakrishnan. He got the Nobel Prize Chemistry 2009. So the most recent Indian to get a Nobel Prize in case of science is your Venkatraman Ramakrishnan. Venkatraman Ramakrishnan. Indian got Nobel Prize in case of economics much later also. But if you take into account science, the last Indian to win a Nobel Prize in the area of science is Venkatraman Ramakrishnan, and he studied the ribosome. The ribosome translates the genetic information to action. The three Nobel Prize laureate in chemistry 2009, Adayona, Stees and Venkatraman, rewarded for mapping ribosome, one of the cell's most complex machineries. So basically, the encoded information is converted into the functional system by what? By the ribosome. Okay. Now, all cells divide. There are two major type of cell division: mitosis and meiosis. Now, mitosis happens in normal cell, our skin cell, liver cell, every time it is dividing. Mitosis occurs in normal cell or somatic cell. Meiosis occurs in the gametic cell, that is the sperm and ova, germ cell, sperm and ova. In case of mitosis, when the cell division takes place, the daughter cells have same chromosome number. So the chromosome number remains same. Whereas in case of meiosis, chromosome number is reduced to half. Okay. In case of mitosis, the DNA of the daughter cell or the two cells which are formed are clone of each other. Their DNA is 100% same. Whereas in case of meiosis, a phenomenon called crossing over and recombination takes place. So the chromosome in pair arrange themselves, then they exchange certain DNA, certain so one of the arm of the chromosome exchange genetic material, and then we have formation of the sperm and ova. That is why each sperm and ova is unique. In case of mitosis, all the cells are same. In case of meiosis, due to there is process of crossing over and recombination between the chromosome pairs, the chromosomes in the pair. So first pair, you have one chromosome 1 and 2, second pair, third and fourth. So first and second chromosome exchange their some of the DNA. Similarly, third and fourth exchange some of the DNA. So new combination comes, and this leads to variation. So basically, evolution works on these variation. So you have two type of cell division, mitosis and meiosis. Meiosis is also called reduction division. So in case of mitosis, all cells have 46 chromosome. In case of sperm and ova, the number of chromosome is how much? 23. So after this, then fertilization takes place. Sperm has 23, ova has 23. So again, when 23 fuse, the zygote has 46. So if meiosis was not happening, then in every generation the chromosome number would have doubled. Okay, so that is about cell biology. Now going into let's start digestive system. So basically, digestion, complex food is broken down into simpler particles. That is the concept of digestion. See, protein is not directly absorbed by the body. Protein is broken down into amino acid. So amino acid is absorbed by the body. Complex sugar will be broken down into monosaccharides, simple sugar, then it will be absorbed by the body. Now, the digestion consists of two things: mechanical process and biochemical process. So, both you have mechanical and biochemical process. For example, teeth. Teeth can break down food particles. Teeth can break down the food particle, that is a mechanical process. And biochemical means use of enzymes. Okay. So, we take various kind of nutrient. Some nutrient are required in larger amount, that are called macro. For example, protein, fats, carbohydrate that we need in bulk is called macronutrient. Some of the things we require in small amount is called micronutrient. Vitamins, minerals, etc. called micronutrient. Fat and carbohydrate, they provide energy for the cell for our daily function. Carbohydrate and fat in bulk provide you the energy for different kind of functions. So they are called energy giving food. Protein are essential component of the cell for muscle and other formation. So these are called bodybuilding food. Protein is called what? Bodybuilding food. Okay. Say in case of a young growing child, we increase his protein consumption. Why? For muscle formation. Or if a person is going for gym or in sports where more muscle is needed, protein consumption has to be increased. Now, for testing protein, we use this test called where we add copper sulfate and caustic soda. So you crush the thing, then you add copper sulfate and caustic soda. And if violet color is there, it indicates the presence of protein. So this is called, this test is copper sulfate and caustic soda for test of protein. And if there is presence of violet color, that is there. In case of starch or carbohydrate, you have iodine test. You add 1 drop of iodine solution, and if it turns blue or black, that indicates the presence of carbohydrate. Just take a look. This is the NCERT diagram. This is your NCERT diagram. These are sources, some of our daily carbohydrate requirement. Rice, wheat, maize. Many of the fruits also provide you sugar and other, sweet potato, etc. These are some of the sources of fat. Fat sources there can be two kind of sources, animal source, plant source. Both for carbohydrate, protein as well as fat. It can be fat, it can be animal source or plant source. In case of fat, you dry it, you take the thing and then wrap it in a your tissue paper. If oil comes on the tissue paper, that indicates the presence of oil. Okay. Now going into the chemical aspect, in case of protein, beside carbon, hydrogen and oxygen, you have nitrogen. Remember, nitrogen is found in protein. Nitrogen is not found in carbohydrate. Nitrogen is not found in case of fat. So fat are made up of CHO, carbon, hydrogen, oxygen. Now fat have been asked more in the recent time due to increasing lifestyle diseases. So fats, carbon has the property of catenation or forming long chains. Carbon has the property of catenation or forming long chains. Okay. So this is the property of catenation, long chain. Now fat can be saturated or unsaturated. Saturated fat have single chain between the carbon atom. Unsaturated fat have double and triple joint between the carbon atom. So if double and triple bond are there, this is unsaturated. Oxidation of unsaturated fat is easy. So there will be oxidized, less deposition will take place in the body. And therefore, they are now considered to be safer for the body, unsaturated fat. If you have one double bond or triple bond, it is called MUFA. If you have two or more, it is called PUFA, polyunsaturated fatty acid. One monounsaturated fatty acid, two or more polyunsaturated fatty acid. Saturated fat, single bond between the carbon chain. Saturated fat, not easily oxidized. Those oil which have more amount of saturated fat will become solid. These are solid at room temperature. So in winters, those oil which become solid have more amount of saturated fat, less amount of unsaturated fat. So if more amount of unsaturated fat is there, it will remain liquid. There is a specific type of unsaturated fat called omega fat. These have been into a lot of news and also in question. Omega fat are PUFA. They are considered good for health. So nowadays you will find this coming into picture. People are increasingly consuming this seed. Flax seed is one of the richest source of omega-3 fat, remember. And fish is considered Best among the non-veg, all the mutton, chicken and fish. Fish is better because fish oil has omega-3 fat. So nowadays you will find people are becoming conscious about omega-3. Lot of health benefit right from eye, reduced cardiac disease, allergy. All those benefits are there in case of omega fat. So that is why omega fat is coming into picture. Less cardiac disease, allergy, mental disorder, arthritis, eye. You have reduced occurrence of disease like Alzheimer and other. So nowadays even in festival you will find people giving packet of seed. Seeds, some of the seed mixture are rich source of omega-3. Then you have one fat called cholesterol. See, people go for testing their cholesterol level. It is normal or higher side. Cholesterol is required. See, it is not, body means balance. So cholesterol is needed for these. It has been asked in multiple exams. Your vitamin D synthesis occurs from cholesterol. Steroid hormone formation from cholesterol. Okay. Reserve source of energy. Remember, lipid is one of the component of plasma membrane or cell membrane. Okay. Now, cholesterol can be three types. HDL cholesterol, high density lipoprotein cholesterol, that is considered good cholesterol. So, there is a test called lipid profile test. There is a test called lipid profile test. In lipid profile test, basically we measure these three kind of cholesterol: HDL cholesterol, LDL cholesterol and VLDL cholesterol. So, HDL cholesterol is considered good. They are good for health. LDL and VLDL are considered not that good. So, if VLDL and LDL are on the higher side, the person needs to be careful. So, remember, good cholesterol is HDL, bad cholesterol is LDL and VLDL. This has been asked by UPSC in many exams. Now, there is a lot of use of Banaspati oil. So, there is a process called hydrogenation. There is a process called hydrogenation. You remember dalda and other Banaspati. So, they become solid. They become solid. So, there is an industrial process called hydrogenation. See, each oil has a particular aroma. Now, what was done? The vegetable oil was from different sources boiled and hydrogen gas was passed through it. So, when hydrogen gas passes through it, the double bonds are broken and hydrogen gets attached. This process is called hydrogenation. Now, if the hydrogen are attached on the same side, this is called cis configuration. And if they are attached on the opposite side, this is called trans configuration. So, trans fat, they were used extensively in your bakery industry, food industry. But what happened? They led to a lot of health condition. It makes oil solid, longer shelf life of bakery product, but they are considered to be bad. They have increased the incidence of your cardiac disease, lot of death due to cardiac disease. Then they lower the good cholesterol and increase bad cholesterol, increase the incidence of diabetes, and that is why WHO had come out with a program called Replace. So the target is to reduce industrially produced trans fat to zero. Some of the trans fat, natural trans fat, occur in animal product like meat, milk, butter. Some amount of natural trans fat can be there. So in India, you have the regulatory body called FSSAI. So by 2022, oil cannot have more than 2% trans fat. Cannot have more than 2% trans fat. Okay. So this is the regulatory body. Now just take a look at, this is the regulatory body called FSSAI for food. So they have the act called Food Safety and Standard Act 2006, based on which this body called FSSAI has been set up. This is something about the applied aspect of nutrition, how government is regulating the food sector. FSSAI has several powers: set the standard of food, what is whole milk, what is skimmed milk, what is toned milk, registration of food business, how the labeling has to be done. FSSAI licenses taken by all the parties which import food, surveillance, safe food management practice, registration, all those aspects are now regulated by this body called FSSAI. It comes under Ministry of Health and Family Welfare. Now, moving back into the digestion process. Now, the digestive tract starts from mouth and ends at the anus. Now, this is the mouth. From mouth, it moves ahead. Here you have pharynx. In mouth, you have teeth. From pharynx, the things move to stomach. From stomach, it goes to duodenum. This is the first portion of small intestine, duodenum. Then it moves. Duodenum is the first portion of small intestine. Then into jejunum. This is the second portion of small intestine. And then ileum. So jejunum and ileum are the second and third portion of small intestine. Then small intestine joins where? It joins the large intestine. So small intestine and large intestine, the classification is basically based on what? The width of the tube. Now, here you have a structure called appendix. This is a vestigial organ. Sometimes people have pain in appendix, so that can be removed. Now, from first mouth, also pharynx, then through esophagus comes to stomach, then small intestine, three portion, first portion duodenum, jejunum, then ileum, then food moves into large intestine, first portion is cecum, attached to cecum is appendix, then ascending colon, it is going up, then transverse colon from this side to this side, then descending colon, it has come down, then finally this is rectum where the food comes, and then finally through anus the food moves out. Okay, so that is their structure. That is how the structure is arranged from mouth to finally the anus. Now, in mouth you have teeth. The teeth are attached in the socket of the jawbone. This condition is called thecodont condition. They are attached to the socket of the jaw. Okay, they are inside the jawbone. The teeth are inserted. In humans, we have two sets of teeth: milk teeth and permanent teeth. That is why this condition, most of the mammals have this condition called diphyodont. Then, in case of human, just take a look at the teeth, upper and the lower jaw. So here you have four different type of teeth: incisor, canine, premolar and molar. The first two are incisor, then one canine, then two premolar, and the last three are molar. The last molar is now considered to be a vestigial organ. It may or may not come out. Okay. Now, attached to this structure are the different digestive glands. In case of mouth, you have salivary gland. Three pair of salivary gland are there: parotid, submaxillary, and sublingual gland. Then stomach. Stomach contains gland. It has the glandular structure on the inner side. Then pancreas, and then small intestine. Okay. So in mouth, the teeth will break it into smaller fragment. Saliva comes. Saliva contains the enzyme salivary amylase and lysozyme. So salivary amylase act on carbohydrate, and carbohydrate is converted into disaccharide, that is maltose. So 30 to 40% of the carbohydrate is digested into disaccharide, that is 2 unit of sugar. Lysosome has antimicrobial action. Then the food moves ahead from mouth through esophagus. The food will finally come to where? Stomach. The food will come to stomach. Stomach has gastric gland. Stomach has what? Gastric gland. It has three kind of cell. One is mucus cell producing mucus. Mucus forms a protective layer because the HCl is highly corrosive. Then peptic cell or chief cell that produce proenzyme pepsinogen, and then finally the oxyntic cell which is producing HCl. Now, the enzyme pepsinogen is produced in a form called pepsinogen. This pepsinogen will be finally broken down into pepsin, which acts on the thing. Okay. So this pepsinogen, on exposure to HCl, when it comes in contact with HCl, then it is converted into the active form called pepsin. So carbohydrate digestion started in mouth, protein digestion starts in stomach. This pepsin convert protein into proteases and peptones. Then the other enzyme which is found there in stomach is renin. Renin acts on milk and milk is broken down. Milk has a protein called casein. This casein is converted into paracasein. So renin enzyme acts on the milk protein or casein. Small amount of lipase. Lipase, the name itself suggests it is acting on a thing called lipid. The enzymes are named after like that. Now, enzymes are specific for pH and temperature. The pH in mouth was neutral. So the enzymes of the mouth will become inactive when they arrive at stomach. Why? Because the stomach pH is highly acidic, 1.5 to 2. The pH of stomach is highly acidic, that is 1.5 to 2. That is why the enzyme of the mouth will stop functioning. So you have pepsin, renin and small amount of lipase. Okay. So basically protein digestion starts from stomach. Small amount of nuclease is there acting on the nucleic acid. Then from stomach the food moves ahead. From stomach the food moves ahead. Okay. Stomach the food moves ahead. And here you have the first portion, that is duodenum. On duodenum you have out toward the inner loop of the duodenum you have the pancreas, from where pancreatic juice will come, and toward the outer side you have the liver and gallbladder. So gallbladder stores bile. Now moving on to the from stomach the food moves into what? Duodenum. That is the first portion. First portion of small intestine. So gallbladder will release bile. Sometime you have stone in the gallbladder, so the sometime the gallbladder is removed and the bile duct is now attached to the duodenum, so that continuous flow of bile takes place. So bile contains the bile pigment. One of the main function of bile is to make the medium alkaline. So the enzyme of stomach will stop acting. And second is emulsification of fat. The fat is broken into smaller particles. So it becomes one, it makes the medium alkaline. So next set of enzyme will take place and the fat is broken into. Then the pancreas will release the pancreatic juice and it contains certain enzymes. The enzymes are trypsin, chymotrypsin, lipase and nuclease. Trypsin and chymotrypsin again act on protein and the protein are broken into smaller fragment called peptones and peptides. Peptones and peptides. Lipase act on lipid. Lipid is broken into fatty acid and glycerol. Nuclease on the nucleic acid. Then the food moves ahead in the small intestine. The second and third portion of the small intestine are jejunum and ileum. Okay. They release intestinal juice. The intestinal juice has certain enzymes. Erepsin. Now the protein has been broken down into peptide and peptone. So erepsin acts on peptides and protein and peptones. Peptides and peptone, and that is finally broken into what? Amino acid. Then you have sucrase. Sucrase acting on sucrose, that is the cane sugar. It is broken into glucose, fructose. Lactase acting on lactose, that is the milk sugar, that is broken into glucose plus galactose. Maltose will be broken into 2 molecule of glucose. So that is how things takes place. So basically protein into amino acid, complex sugar into monosaccharide, that is single sugar, fat into fatty acid and glycerol. Now in small intestine, absorption of nutrient also take place. These are the structure. The inner lining of small intestine is folded into finger-like projection, and these finger-like projection are called villi. These finger-like projection are called villi. They increase the surface area. They increase the surface area for absorption. So these are called villi. The digested nutrients are absorbed by them. Now just take a look how the absorption takes place. This is from your NCERT. So in mouth, certain drugs are absorbed. If you remember, certain drugs in homeopathy and other, they give into your mouth because mouth is richly supplied by blood vessels. So just inside on the lower portion of the tongue, the drug is given, it is absorbed. Stomach, absorption of water, simple sugar, say you take glucose, that would be absorbed in the stomach. Alcohol is also absorbed in stomach. Small intestine is the principal organ for absorption. Digestion and absorption principally takes place in small intestine. In small intestine, you have digestion of nutrient. So the digested nutrient are what? Glucose, fructose, fatty acid, they are amino acid, they are absorbed in the small intestine. Okay. And in large intestine, you have absorption of water, mineral, and certain drug. In large intestine, you have absorption of water, some mineral, and drugs. One of the important gland is liver, largest gland, asked multiple of time number in exam. Then it produces bile. Bile is the excretory product of hemoglobin metabolism. Urea formation occurs in liver. This has been asked by in many exams. Urea formation does not ever happen in kidney. So here people make an error. In many exams, this question has been asked. Urea synthesis occur in liver, excretion is through kidney. Then an important component of blood is fibrinogen. So in clotting, fibrinogen changes into fibrin. So that is there. So fibrinogen has a role in clotting. Then heparin. Heparin is found inside the blood vessel. It prevents clotting. Detoxification of drug and chemical happens in liver. In case of alcohol consumption, basically alcohol has to be converted into harmless form, that is to be detoxified. That is why in case of alcoholic people, liver damage is there. Liver damage is there. And it is also a storage organ. Just take a look at some of the sources where you have the vitamins, micronutrient. Papaya, carrot, milk, fish oil, mango, they have source of vitamin. Vitamin B, we say whole grain should be consumed. Meat, whole liver, whole grain are very rich source of vitamin B. Vitamin C, ascorbic acid, these are the sources. Chili, citrus fruit, guava, tomato, these are rich in what? Vitamin C. Vitamin C deficiency called scurvy. Then some sources of iodine, just take a look at. And in animal source, you have the aquatic sources are rich source of iodine, spinach, then your ginger, some sources of phosphorus. Take a look. Vitamin A, if deficiency is there, you have night blindness. Vitamin B1, very weak muscle. Vitamin C, bleeding gum on scurvy. Vitamin D, rickets, deformation in bone. Calcium and iodine, calcium is required for bone and tough tooth. So in a growing child, the requirement for calcium would be more. Iodine deficiency causes goiter, that is enlargement of the gland, thyroid gland. Iron deficiency causes anemia. Iron deficiency causes anemia. Then moving on to circulatory system. So the purpose of circulation is transport. The transport system of the body is circulatory system. So from one part of the body to different part of the body, digested nutrient, biomolecule, hormone, antibodies, heat. So if you are doing exercise, say if your hand muscle is working, the heat will also be transferred to your leg and back. So it transfers various things, nutrient, hormone, waste product, heat, etc. throughout the body. So circulatory system has a pumping organ, that is heart, artery and veins. In general, artery carry oxygenated blood, vein carry unoxygenated blood. This is the circulatory system of human. So the blue one represents what? Unoxygenated blood, and the red one oxygenated blood. Heart has 4 chambers in mammal. The upper chamber are called auricle or atria. The lower chamber are called ventricle. So impure blood represented by this blue color comes to right auricle. This is the right atrium or right auricle. When the heart compresses, the blood moves from right auricle to right ventricle. Now when the ventricle presses, through right ventricle, it goes to, through pulmonary artery, it goes to lungs. So one exception is pulmonary artery, only artery carrying impure blood. Now in lungs, what happens? CO2 is given out, oxygen is taken in. Now the oxygenated blood through pulmonary vein, this is again exception. This is again an exception. Through pulmonary vein comes back to left auricle. From left auricle, it goes to left ventricle when it compresses. And from left ventricle through aorta, it goes to the body. So that is how the cycle runs. Impure blood comes to right auricle to right ventricle. Right ventricle, it goes to, through pulmonary artery, it goes to lungs. From lungs, exchange of gases take place. Oxygenated blood comes back to left auricle to left ventricle, and from left ventricle to the body through aorta. That is how it takes place. Now, heart is a autonomous, semi-autonomous organelle because its electrical system is different. So, even if the brain is dead, the heart can vibrate. Heart has specific area called a point called SA node or pacemaker. Take a look. That was the structure of the heart. This one is the pacemaker, SA node. This is AV node. So, SA node, AV node, then you have bundle of His and Purkinje fiber. That is how the electrical system of the heart works. So, heart, the SA node or the pacemaker generates an electrical impulse. First, it goes to the upper portion, that is the auricle. Then through AV node, it goes into the ventricle, both sides. So, first your auricle will contract. After a gap, the ventricle will contract. So, the heartbeat or the electrical impulse for the heartbeat start from SA node. So, this is also called pacemaker. So, SA node is the pacemaker. So, this pacemaker generates electrical impulse which causes the heart muscle to contract. Sometime in some people, the natural pacemaker is not producing sufficient amount of electrical impulse or electrical current. In that case, we go for artificial pacemaker. So, here the medical device assists the electrical current formation. So, this is your artificial pacemaker. Now, in case of cardiac disease, there is a very common technique called ECG, electrocardiography. Here, recording of the heart, electrical activity of the heart is done. ECG, electrocardiography. Basically, recording of the electrical impulse of the heart is done. So, like this, PQRS complex is there. So, a expert doctor, by taking a look at this ECG report, can find out what is the problem, in which component the problem is there. Okay. Thank you. Thank you. Okay, in which component the problem is there. So, very common technique, that is ECG. Nowadays, say ECG is normal. There is another test called treadmill test. In gym, you have treadmill. So, basically, treadmill test. In treadmill test, the person is made to walk on a treadmill at increasing difficulty. Say 2 km per hour, 4 km, 6, 8. Like this, they will increase their difficulty, and in the same way, they will reduce their difficulty. And in the entire process, the ECG, electrical activity of skin, rate of breathing, etc. are measured. Heart rate, BP, all the parameters are. This is called treadmill test or TMT. So this is called stress electrocardiogram. Beside ECG, other parameters are also monitored. This has become very important. Treadmill test. So basically, it shows how the heart function under stress. We just discussed about an artificial pacemaker. So basically, it is a battery system placed under the collarbone. The electrical lead is taken and it is connected to the point of your SA node, where the natural electrical impulse was generated. So the SA node is assisted by what? It is assisted by the pacemaker. The natural SA node is assisted by the pacemaker. Now, there are certain diseases of the heart. We'll come into that. There's an artery called coronary artery. The heart is a muscular organ that functions 24/7. The heart is continuously vibrating, so it also needs energy. So for energy, it needs nutrient, and second is oxygen. So basically, glucose is oxidized to produce CO2 and ATP. So the arteries that supply blood to the cardiac muscle are called coronary artery. They are called coronary artery. Now, sometime what happens? Fat get deposited inside the coronary artery, and the lumen or the passage available for movement of the blood is restricted. So fat gets deposited on the wall of coronary artery, and it can lead to heart attack and other condition. So coronary artery supply blood to the cardiac muscle. If the fat gets deposited there, what happens? The passage is reduced. The passage is reduced. So that is why there is a test called angiography. Angiography gives idea about the percentage of blockage in the coronary artery. There is a technique called angiography. Angiography gives idea about the percentage of blockage in the coronary artery. Now there are two methods to overcome it. One method is called angioplasty, where a stent is placed in this narrowed artery. Take a look over here. Here fat is deposited on the wall of artery. So here what is done? In angioplasty, a thin stent is or a wire is put through the veins, through the artery or veins in the leg or hand, and it is carried into the heart. And then over the wire, a balloon and a stent is taken. The balloon is inflated and the wire mesh or the stent is placed at this point. So it opens up the narrowed artery. It slightly opens up the narrowed artery. And if blockage is very high, bypass surgery is done. So you take a vein from that person body and you attach it across the point of blockage. That is your bypass surgery. Say this is the heart. Now this coronary artery is blocked more than 90%. This coronary artery is blocked more than 90%. So what is done? A vein from the leg is taken out and surgery is done and it is attached across the point of blockage. So this is called bypass surgery. This is called bypass surgery. So earlier a lot of cut was required. Now you have robotic and other system coming into picture. So one aspect is angioplasty. You place a stent. Second aspect is bypass surgery. Second aspect is what? Bypass surgery. Then we have a condition called heart attack. So when heart attack happens? Heart attack happens when flow of blood to the heart muscle is blocked and some of the heart muscle die. So the heart muscle continuously require oxygen. Now If the artery is blocked or due to any other reason, the heart muscle do not get sufficient amount of oxygen, the heart muscle die. This condition, there should be death of the cardiac muscle, and then only it will be the case of heart attack. Otherwise, it is called ischemia. Now, there is a concept called brain death. A person heart can be vibrating, but he becomes brain dead. So, if there is an accident, the person is put on ventilator. So, on ventilator, the breathing is supported from outside, and by the time the brain dies, the brain becomes dead. Brain has three portion. Brain has three portion: forebrain, midbrain, and hindbrain. So, if the midbrain and hindbrain together are called brainstem. In India, brain death is the case where brainstem is death. ठीक है? You have act called Transplantation of Human Organ Act, which has allowed organ donation from live and brainstem dead donors. So, when the brainstem becomes dead, the person is brain dead, but his heart is vibrating. Okay. So, his heart is vibrating. In a case of normal death, cardiac death, only cornea and skin can be donated. Whereas in case of brain dead person, multiple organs can be donated. Okay, just take a look over here. In case of natural death or cardiac death, only cornea and skin. Whereas in case of brain dead person, the heart is vibrating, the person is on ventilator. So brain, he is dead, he is brain dead. So multiple organs can be harvested. And for that, government has set up an organization called NOTO, National Organ and Tissue Transplant Organization, to streamline the entire process. So a brain dead person can supply multiple organs: heart, lungs, pancreas, two kidney. So technically, a brain dead person, if the family member agrees, he can give life to 6 to 7%. Now coming to the cardiac system, one is the heart and second is the blood, the body fluid. Blood has two things. The liquid portion is plasma and second is cells. So 55% of the portion is liquid, that is plasma. If you put blood in a test tube, the liquid portion remains on the upper side, the cell will settle down. So 55% is the liquid and 45% is the cell. In this plasma, you have different protein. Primarily, plasma is your water. Along with that, salt and other proteins are there. Three main proteins are there: fibrinogen for clotting of blood, globulin for defense mechanism, and albumin, which maintains the osmotic balance. So albumin is also found in egg. So albumin, the egg white is albumin. The egg white is what? Albumin. You have three kind of cell: RBC, WBC, and platelets. So one is the plasma and second is cell. RBC contains the pigment hemoglobin. RBC has the pigment hemoglobin. Now, to increase the efficiency, the nucleus is thrown out and the entire space is filled with what? Hemoglobin. So, nucleus is absent in mature RBC. It is not there. Now, in male, you have 5 million RBC per millimeter cube, and female it is 4.5 million per millimeter cube. RBC formation occurs in red bone marrow and destruction in spleen. Spleen is also called graveyard of RBC. Spleen is called what? Graveyard of RBC. Okay, so RBC lacks nucleus, mature RBC don't have nucleus. Main function of RBC is what? Transport of oxygen. Oxygen combines with hemoglobin to form oxyhemoglobin. Then you have white blood cell or the leukocyte. These are white blood cell. There are different type of leukocytes. White blood cell contain nucleus. Their number is 5,000 to 10,000 per millimeter cube. In case of disease condition, the number of white blood cell will go up. White blood cell can be divided into two broad groups: If granules are present, granulocyte. If granules are absent, agranulocyte. Then staining is done. Stains are done. If it takes acid color, it is called eosinophil or acidophil. If it takes basic color, it is called basophil. And if it takes neutral color, it is called neutrophil. Now, the maximum number is of neutrophil. 60 to 65% of all the white blood cells are neutrophil. They are the first line of defense. They are the first line of defense. When any infection enters the body, the first to go and fight is your neutrophils. Basophils are less. They secrete compound like histamine and other which cause inflammation. So the area is swollen up. Histamine, serotonin and other, they cause inflammatory response. Eosinophil, their number increases in case of allergy or certain specific condition. That is why nowadays you go for CBC, complete blood count. So if neutrophil and others are increased, there is a chance of infection. If eosinophil has increased, it is indication that you are suffering from allergy. Allergy can be due to various aspects. Then one was your neutrophil, one was your granulocyte, other was agranulocyte, where no granules are there. In agranulocyte, important is lymphocyte and monocyte. Lymphocyte, you have two types, B lymphocyte and T lymphocyte. B lymphocyte produce antibodies and T lymphocyte are responsible for your memory, the memory of the immune system. You have different type of T lymphocyte and one of the T lymphocyte is helper T cell. In case of HIV or AIDS, the number of helper T cell is reduced, remember. So HIV, in case of HIV infection or AIDS infection, basically the helper T cell is reduced. So the immune system breaks down. AIDS, acquired immunodeficiency syndrome, the immune system breaks down. Largest WBC are monocyte. So their number increases in case of tuberculosis. So basically based on the difference that is there. Then you have RBC, WBC, the third is platelet or thrombocyte. Platelet or thrombocyte. Their number varies from 1.5 to 4 lakh per millimeter. If you remember a very common disease called dengue. In case of dengue, the platelet is reduced. The role of your platelet is primarily in blood clotting. So whenever you have injury or cut, these come to the site of injury. They release prothrombin. In place of calcium, the prothrombin is changed into thrombin. So these platelets release prothrombin. Prothrombin in presence of calcium is changed into thrombin. Now thrombin acts on the soluble protein called fibrinogen. Fibrinogen, which was made by liver, is soluble protein. Now what happens? This calcium and thrombin converts fibrinogen into fibrin and the fibrin threads come out of the blood and they other component of the blood get deposited there and that is how the clot forms. So if the number of platelet is reduced, clotting will be affected. And this is there in case of dengue. In case of dengue, the blood clot comes down sharp. Now coming to blood group, you have main blood group A, B, AB and O. Now this is based on two things. One is the antibody or proteins which are present on the surface or glycoprotein present on the surface of RBC and antibody present in the plasma. Now in case of type A, the antibody is there of A type. Antigen is type A that is present on the surface. And antibody is found in plasma. Antibodies are produced by B cell. They are present in the Y-shaped molecule. They are present in the plasma. So in case of A group, you have antigen A and antibody in the plasma is against B, anti-B. Group B, antigen is B antigen and antibody is against A. So if you give B to A, this anti-B will react with B and immediate clotting will happen. So same should be given. In case of AB, there is no antibody. They have both antigen A and B, but they have no antibody. They have nothing against either A or B. That is why AB is called universal acceptor. It accepts all the blood group. In case of O, there is no antigen on the surface, neither A antigen nor B antigen, but it has antibody against both A and B. So if O cannot receive blood from anyone, O can be donated to all because it has no antigen, so it will not react with antibody. O is called universal donor, but O will receive blood only from O, only from O group. Okay. Then coming to endocrine system. So in our body, you have two type of gland, endocrine gland and exocrine gland. Endocrine gland produce hormones, and these hormones are carried by blood. They do not have any pipe or duct. That is why these are called ductless gland. Endocrine gland do not have duct or pipe. They are called ductless. There is no pipe. So endocrine gland produce hormones. In digestion, we had read about enzymes. So there is difference between enzymes and hormones. Let us come to the difference. First difference is one is produced by the hormone is produced by ductless gland, whereas enzyme are produced by glands that have duct. Number two, chemical nature. Hormones can have varied chemical nature. It can be steroid, it can be protein, it can be iodinated amino acid. So there can be various chemical nature. Whereas in case of enzyme, they are proteins. So chemical nature is variable. Number third, enzymes have local action. If you remember in digestion, the enzyme action is very specific. The enzyme of mouth will not act in stomach. Why? In mouth, the pH is neutral, in stomach, it is acidic. Whereas in case of enzyme, they have action throughout the body. Sorry, hormone have action throughout the body. Enzyme have localized action. Enzymes have localized action. Hormones have action throughout the body. Now, these are the important glands. So, associated with brain is hypothalamus, pituitary, and pineal gland. Then in your neck, you have thyroid and parathyroid. Then thymus gland near the heart, pancreas in the infold of your uh, the first portion of small intestine, that is duodenum. Then above kidney you have adrenal gland, and then you have ovary in female and testis in male. Ovary in female and testis in male. So the first gland, let us come to that is hypothalamus. Now this is considered with the master gland. Earlier pituitary was there, this is the actual master. Part of brain. Why it is the actual master? Because it regulates the anterior pituitary. The front portion, if I am standing like this, front portion is anterior. Regulates anterior pituitary, the hormone production from anterior pituitary. So it produces hormones called neurohormones. What it produces? Neurohormones. Okay. Neurohormones. Because basically brain is made up of neuron, that is why the term neurohormones. So it can be two types. One is to increase production, that is called releasing hormone. Another is to suppress production of hormone, that is inhibiting hormone. Releasing hormone, inhibiting hormone. Okay. More beside that, one is it is regulating anterior pituitary. Beside that, two hormones are produced in hypothalamus, oxytocin and vasopressin, which is then carried down to pituitary. Now just take a look over here. This gray color structure over there is hypothalamus, and attached to hypothalamus is pituitary. Pituitary has two portions, anterior and posterior. The two hormones vasopressin and oxytocin are produced in hypothalamus but carried to posterior pituitary. And hypothalamus produces releasing hormone and inhibiting hormone which regulate anterior pituitary. So anterior pituitary is regulated by your hypothalamus. That is the actual master. So attached to the lower portion that in the diagram we discussed, it has front portion anterior, posterior. So first come to the posterior pituitary. In the posterior pituitary, both the hormones were produced where? Both the hormone of posterior pituitary are produced by hypothalamus. And the hypothalamus, oxytocin and vasopressin carried to posterior pituitary. Now the first hormone is oxytocin. This is called childbirth hormone. At the time of childbirth, oxytocin causes contraction of the uterus. The fetus is growing inside the mother's womb, that is the uterus. So it causes the contraction of uterus muscle and then childbirth takes place. It is called childbirth hormone. It is also called milk let down hormone. It causes contraction of the myoepithelial cell of the mammary gland. So the milk is released from the mammary gland. That is why it is called milk let down hormone. And that is why this was misused for milking of the cow at any time they wish. The animal husbandry, what happens? They started using this hormone, injecting the cow with oxytocin hormone and then milking the cows and buffalo. So the first misuse of this hormone, large scale misuse of this hormone happened for milking of cow. Then again misuse was there for increasing the size of vegetables and fruit. So somehow by serendipity or whatever may be the factor, dilute oxytocin is injected into the fruit and vegetable and their size increase overnight. So that is how this hormone is entering into the food chain and threatening the healthcare system. So many a time, lot of discussion has happened how to regulate it, to do all about that, prevent the misuse. That is why remember this oxytocin is important. So oxytocin is called childbirth hormone, milk let down hormone. Lot of misuse of this hormone has taken place. Second hormone from posterior pituitary is vasopressin. In kidney, blood is filtered, then from that filtrate, useful product and water are reabsorbed. So daily our kidney filters around 200 liquid filtrate come out, but actual urine is 1.5 to 2 liters. So 99% of the water is reabsorbed. So this reabsorption is done under the influence of this hormone called vasopressin. Now if there is deficiency of vasopressin, this water reabsorption is less, so excess amount of urine is produced, and this condition is called diabetes insipidus. Excess urine production is there, but urine does not contain your glucose. Urine does not contain glucose, and this condition is called diabetes insipidus. The diabetes mellitus is where sugar has urine has sugar. Here excess urine is there, and you have but there is no glucose present in the urine. Now coming to anterior pituitary hormone, the first hormone is growth hormone or somatotropin. The name itself suggests it is regulating what? Growth and development. So this causes elongation of bone, formation of muscle, etc. So now when a child is growing, he must have sufficient amount of this hormone. Okay? So the child must have sufficient amount of this hormone. And if the hormone is secreted less, it leads to a condition called dwarfism. And if more secretion is there, that is called gigantism. The person grows. Excess secretion, gigantism; less secretion, dwarfism. Now, since it helps in muscle formation, that is why many of the sports people have misused this hormone to increase the muscle mass. So that is why this hormone has been misused in doping purpose. Then thyroid stimulating hormone. Second hormone from pituitary is, see, earlier this was considered to be the master. Why? TSH, thyroid stimulating hormone, is regulating the thyroid gland. So it controls, but anterior pituitary is itself controlled by whom? Hypothalamus. So actual master is what? Hypothalamus. So hypothalamus message come to anterior pituitary and then anterior pituitary to the glands. Name itself TSH. It activates or regulates thyroid gland. ACTH, adrenocorticotropic hormone. This regulates the adrenal gland. Adrenocortic adrenal gland, I will just show you. This is the gland found over kidney. The outer portion is called adrenal cortex. ACTH regulates this adrenal cortex. Then follicular stimulating hormone FSH. In female, the egg is present in immature form in the ova. So under FSH, immature egg changes into the mature egg. LH, release of egg. Maturation under FSH, release under LH. Milk releasing hormone is oxytocin. Milk formation happens under the hormone influence of another hormone called prolactin. Okay. Then thyroid gland. This is the largest endocrine gland found in the neck area. It produces two hormones, tetraiodothyronine or thyroxine and triiodothyronine. Tri means 3, tetra means 4 iodine molecule produced. So this regulates the basal metabolic rate of the body. So this hormone has what? Iodine. Tetraiodothyronine and triiodothyronine. And if there is deficiency of iodine, there is enlargement of the thyroid gland, and this condition is called goiter. The neck area becomes enlarged if there is deficiency of iodine. Another hormone from there is calcitonin. So calcitonin is another hormone produced by thyroid. It causes, see, when you take milk, calcium is absorbed from milk, it goes into blood. Then finally, it can, when surplus or excess is there, this calcium will be deposited in the bone. So calcitonin causes deposition of calcium in the bone. And there is another endocrine gland called parathyroid. Just inside the thyroid gland, you have 4 small parathyroid glands. Parathyroid glands are embedded in the thyroid gland. These 4 red colored dots are parathyroid, and this large lobe is thyroid. So parathyroid gland produces parathormone. So calcitonin deposits calcium in the bone and parathormone removes calcium from the bone. One causes mineralization of the bone, other causes demineralization of the bone. So depending upon your calcium supply, calcium can be deposited in the bone or calcium can be withdrawn from the bone. And these two hormones there are calcitonin and parathormone, which are opposite to each other. And that is how they maintain the balance of calcium. Then you have this gland in brain called pineal gland. Pineal gland, you have this, there is another gland called in the brain portion is pineal. Now, this gland produces a hormone called melatonin. This regulates the body's diurnal rhythm. Our body has a rhythm. It is adjusted according to the 24-hour cycle. It is adjusted according to the 24-hour cycle. You have a time of waking up, sleeping, fecal defecation. Every person has a time. So body 24-hour clock is regulated by this hormone. Sleep, body temperature, other aspect of metabolism. So when people are traveling from one country to another, sometimes this body clock becomes affected. Thymus gland. This is present near heart and the lymphocyte mature out here. You have T lymphocyte and B lymphocyte. So this is located near the heart. The lymphocyte, T lymphocyte mature near the thymus. Now, in old age, the thymus uh, disappears. It digresses and finally it is absent in old age. The thymus gland goes away or disintegrates and it is there. So, in old age, the immunity is lower than a young person. Why? Because the T cell the T cell undergo maturation in the thymus, and in the old age, this is degenerated. Since it is degenerated, the cells cannot, the immunity of a old age person is less than a younger person. Then come to pancreas. Pancreas is an organ which produces hormone as well as enzyme. Pancreas has role in digestion also. Now just take a look over here. There are two portions of pancreas: exocrine portion which produce enzymes, and enzymes are taken by duct or pipe, whereas the hormone producing area is called endocrine or islet of Langerhans. And here the hormones move through what? Blood. So we talked about endocrine glands are ductless gland. So pancreas has two portions. One portion produces hormone, another portion produces enzyme. The enzyme producing area of the endocrine portion is islet of Langerhans. Islet of Langerhans. Two important hormones are produced by pancreas. One is insulin, another is glucagon. Insulin and glucagon. Insulin is made by beta cell, and this converts glucose to glycogen. Another is glucagon, which breaks down glycogen into glucose. Say you have taken your meal. Now glucose is absorbed. Now glucose level in your blood rises. So at that time what happens? Insulin comes into picture and glucose is converted into glycogen for storage. So your blood sugar will come down. So here glycogen is being formed. If there is something is formed, this is called glycogenesis. Genesis means formation. Glycogen is formed. Say after 2 hours, 3 hours, your blood sugar level or blood glucose level comes down. Then your brain is not getting sufficient amount of glucose. In that case, this glycogen will be broken down back into glucose. And here you have role of glucagon. So glucagon will break down glycogen into glucose, and this is called glycogenolysis. This is called glycogenolysis. Glycogen is being broken down. So there they are opposite of each other. There they are opposite of each other. Okay. So there is a disease called diabetes associated with insulin. If insulin is produced less, the blood sugar level will be high. Now this blood is filtered by kidney. Now excess glucose will come out in filtrate, which is not completely reabsorbed, and that is why some amount of glucose will appear in urine, and this condition is called diabetes mellitus. This condition is called what? Diabetes mellitus. So if there are two things, either less insulin is produced or insulin is not acting. Okay, and this condition is your diabetes mellitus, presence of glucose in urine. Vasopressin deficiency was diabetes insipidus. So in these days, you have a lot of cases of insulin diabetes. India is sooner going to become the diabetic capital of the world. Different reports suggesting every 12th Indian is a diabetic or pre-diabetic. Basically, there are three types of diabetes. Type 1, when pancreas is producing less insulin. Type 2, insulin is being produced, but insulin receptors are less on the cell. This is type 2. And third is gestational diabetes. Sometime during pregnancy, the blood sugar shoots up. Diabetes or increased blood sugar itself is not problem, but there are many complications. Why increasing diabetes? Obesity increases the chance of insulin, lack of physical activity, poor eating habit, stress. These are some of the factors regarding increasing incidence of diabetes. Increase aging population. Generally, diabetes happens in after 40 generally. So as the longevity increases, that it could be happen in 60s. That is why people go for testing their blood sugar regularly. Diabetes can lead to lot of complication: kidney damage, nerve damage, retina damage, foot. Five. Sometimes even the entire foot has to be cut. Wound healing is delayed, and that is why diabetes is a major lifestyle disease. So government has come out with this program, National Program for Prevention and Control of Diabetes, Cardiovascular Disease and Stroke. So now more people are dying due to lifestyle disease. Infectious disease are less, lifestyle disease or non-communicable disease are more prominent. Cardiac disease, diabetes, these are example of non-communicable or lifestyle disease. They are not bacterial and viral disease are communicated from one to other. These are not moving from one to other. This is due to either genetic factor or sedentary lifestyle. So that is why to prevent, healthy diet, physical activity, alcohol and tobacco use should be less, stress management, these are the factors. Then the gland over kidney is adrenal. Adrenal gland is found where? It is found over the kidney. Adrenal gland is found over the kidney, adrenal. So it has two portions. The outer portion is cortex, the middle portion is medulla. From middle portion, you have two hormones, adrenaline and noradrenaline. Adrenaline is the emergency hormone. In exam, you have asked multiple times. So basically, these are emergency hormone or under stress. So they increase the heart rate, respiration rate, blood supply, and produce your body for emergency. This is also called triple F. Flight, either you have to move away, fight or fear. Flight, triple F hormone, adrenaline. So adrenaline and noradrenaline have similar impact. They prepare the body for emergency situation. So one is the middle portion producing adrenaline and noradrenaline, and then you have the outer portion or adrenal cortex. Cortex is the outer portion. From here, the hormones are mineralocorticoid. The name itself suggests it is something to do with minerals and ions. So sodium potassium balance, mineral balance in your body is being regulated by what? Mineralocorticoid. And the main mineralocorticoid is aldosterone. Aldosterone is the main one. So remember, two questions asked in many exam. One is your emergency hormone is adrenaline, then mineralocorticoid from adrenal cortex, glucocorticoid regulate the glucose mechanism. They break down lipid and protein, and the glucocorticoid also act as they help in maintaining the cardiovascular system, and they suppress inflammation. So many of the inhalers have cortisols. And last from here, the third hormone from adrenal cortex is adrogenic steroid. So basically, they have muscle building effect, secondary muscle building effect, and they control the secondary sexual character in males. Okay, secondary sexual character like change in voice, formation of hairs on the body, etc. Then in males, you have testis, and female, you have ovary. So in male, testis produces androgen hormone. The main hormone is testosterone. So testosterone are basically from androgen group of hormone, and they regulate the secondary sexual character. Secondary sexual character in male, change in voice, hair on body, muscle formation, then function of additional gland like prostate gland, urethra, etc. Okay, so they have anabolic effect. They help in building up of muscle. That is why this hormone has been large. This testosterone was one of the hormone used in sport for doping purpose. It builds up the muscle mass. So it was banned. Testosterone from animal or recombinant testosterone was banned long back by the International Olympic Committee. From ovary, you have two hormones. One is estrogen. This regulates the reproductive cycle and the menstrual cycle in female. And second hormone is progesterone. Progesterone is called pregnancy hormone. Progesterone helps in the attachment of fetus to the uterus wall. So estrogen regulates the reproductive cycle of the female, whereas if fertilization takes place, the fetus has to be attached to the uterus wall. Attachment of fetus to the uterus wall. Okay, so that is about these endocrine glands. Then coming to respiration or the respiratory system. So all life processes need what? Energy. Energy is in the form of what? ATP. In cell biology, we have read about the mitochondria is the power of the house of the cell where ATP is produced. So basically, respiration occurs where? Finally at the level of cell. You inhale air and other, finally that oxygen goes to the cell and there oxidation takes place. So respiration is a chemical reaction that happens in the living cell. How? Whether it is plant cell or animal cell, all of them require oxygen. Okay, there are certain anaerobic organisms that is different, but in general, oxygen is required, whereby glucose is oxidized to CO2 and you have production of ATP. Now, respiration can be aerobic. In aerobic, the term is air. The term aerobic comes where? From where? Air. So, air contains around 21%, more than 21% oxygen. So, basically, when the air goes in, the oxygen is of the air is utilized. How? Glucose is oxidized to produce CO2 and water and ATP. So, this happens. So, this reaction is what? Aerobic respiration. So, glucose is oxidized to produce CO2 and water. Glucose is C6H12O6. It combines with oxygen to form 6CO2, 6H2O plus 38 ATP. So, ATP is the energy currency of the cell. So, basically glucose is oxidized to produce CO2, water and ATP. So, one is aerobic respiration. Sometime in our muscle also, say heavy work is being done. At that condition, muscle may not get sufficient amount of oxygen, and in that condition, anaerobic respiration takes place. Anaerobic means temporary deficiency of oxygen. In that case, in our muscle, what happens? Glucose is changed into lactic acid, and small amount of energy is reduced. That is why muscle pain or fatigue happens due to accumulation of lactic acid over a period of time if you are doing exercise. So, finally, at the end the lactic acid will be removed and then it will be oxidized again. So, but that is anaerobic. So, one is your anaerobic respiration in humans where you have lactic acid formation, and there is another anaerobic process which is also done by microbes. One type of anaerobic respiration in yeast and unicellular yeast and some other microbe is production of alcohol. See, glucose or sugar is fermented to produce ethanol and CO2. So, under anaerobic condition in absence of oxygen, the sugar is fermented to produce ethanol. Ethanol is C2H5OH. The drinking alcohol is basically ethanol and carbon dioxide. Ethanol and CO2. Ethanol and carbon dioxide. Ethanol is utilized by the brewers or for the brewery purpose, for the drinking purpose. All the form of alcohol, wine, beer, all them are basically ethanol. And CO2 is used for by the bakery process. So in bread also you put yeast, fermentation takes place, ethanol is there and carbon dioxide. Now when the bread loaves are put into the furnace or in the bakery furnace, what happens? The ethanol will evaporate away and that space expands. So that is there. That is why it becomes fluffy. So any glucose, any carbohydrate or sugar can be fermented to produce ethanol. It can be from rice, it can be maize, molasses, sweet potato, even your potato, grapes, fruits. Basically, sugar is fermented into ethanol. That is why sometimes you have the smell like that. Now, in respiration, there are two basic things. First step is two-step glycolysis and Krebs cycle. In first step, the first step of respiration is your glycolysis. Glycolysis occurs in the cytoplasm, inside the plasma membrane. Basically, glucose is broken down into pyruvic acid. 2 ATP is produced and 2 NADH. 1 NADH in future cycle produces 3 ATP. The total 8 ATP is produced in this. Net is 2 and gross is 8. Why? Because 1 NADH in future cycle will produce 3 ATP. So 2 NADH, 2 into 3 is 6 plus 2 ATP. Krebs cycle occurs inside mitochondria. So the chemical process of oxidation is in two steps. In first step, glucose is converted into pyruvic acid. This happens in the cytoplasm and this is glycolysis. Then this your pyruvic acid will further convert and go into the Krebs cycle where it is finally oxidized into CO2 and water. There are many respiratory structures found in different animals. Coming to the respiratory structure, in some organisms, the exchange of gases can happen over the cell surface. For example, bacteria, prokaryote, etc. Here, the exchange of gases on the cell surface. Then, in organisms like earthworm and frog, some amount of exchange of gases also takes place through the wet surface or the skin. This is called cutaneous respiration. That is why you will find earthworm and frog after rain. In dry condition, you will not find, or you will find less number of frog or earthworm over there. Earthworm respires, exchanges gases with the help of its skin also, moist skin. Then in insect, you have tracheal system. You have these opening on the side of the body. The body has many segments. In each segment, two opening is there. And these are called tracheal system. Tracheal system is found in the case of insects. Okay. And in many of the lower animals found in water, in fact, gases can be exchanged through the general surface. Then you have lungs. Amphibia, reptile, birds, all of them have lungs. In lungs, you have alveoli where exchange of gases takes place. Amphibia, reptile, bird and mammal. In frog, lungs is also there and also through skin or cutaneous respiration, both is there. Then fishes have a special structure called gills for exchange of gases. So the water moves through the mouth, it passes over the gills, exchange of gases takes place over the gills, etc. Then you have air sacs. Air sacs are basically extension of lungs. Air sacs are found in birds and they make the body of the bird light. So that is why this helps the bird in flying. So these are basically extension of lungs. Air sacs makes the body of the bird light. This is example of flight adaptation. Now coming to human structure, human respiratory structure. Here you have nose, nasal cavity, then you have pharynx, then larynx, then trachea, then bronchi, and then bronchiole, and finally alveolus. So air enters the nasal cavity. Here you have hairs and mucus, hair and your slimy substance. So it traps the dust particles. Then you have to come to pharynx. Pharynx is the common opening for respiratory system and digestive system. Then from pharynx it goes to larynx. Larynx is the voice box. Now there is a structure called epiglottis. When the food or water is being swollen, the epiglottis will close the larynx. Sometime during when you are taking in food or water, the larynx does not close and the food or water goes inside the your this inside the trachea. All this passage, nasal cavity, pharynx, larynx, trachea, bronchi, bronchiole, and finally alveoli. Exchange of gases take place only at the level of alveoli, nowhere else. Nowhere else. Okay. So larynx is the voice box. We discussed nostril into pharynx, pharynx into larynx, pharynx into larynx, larynx is the sound box, then trachea, then bronchi, then bronchiole. In case of respiration, what happens? First, you breathe in, you take air from outside and also throw out it. Then the oxygen, diffusion of gases, CO2, oxygen will go into the blood and then carried to the body. CO2 has to be thrown out from the blood through the lungs, diffusion of gases. Then third, transport of gases. Oxygen has to be transported from lungs to each and every cell. Now CO2 has to be transported back. Then oxygen is utilized by the cell finally for oxidation of glucose. So glucose has to be oxidized to CO2, water and release of ATP. Now just take a look at this chart or this diagram. This is inhaled air and this is exhaled air. So when you take a breathe in air, it has 21% oxygen and how much is the CO2? 0.04. So when you breathe out, what happens? The CO2 oxygen is used and here what happens? The oxygen amount is reduced from 21 to how much? 16.4, around 16%. Whereas the CO2 amount goes up. So inhaled air has 21% oxygen and only 0.04% carbon dioxide. Exhaled as the oxygen is reduced and CO2 is increased. Now going into how respiration takes place. There are two things, breathing in and breathing out. Breathing in is called inhalation, breathing out is called exhalation. Breathing is called inhalation and breathing out is called exhalation. Just take a look at over here. So how you breathe in or how inhalation takes place. Mammal has a structure called diaphragm. Mammal has a structure called diaphragm. This diaphragm divides our body cavity into two parts, digestive cavity on the lower side and respiratory cavity on the upper side. So lower side you have digestive cavity, upper side you have respiratory cavity. So what happens? Diaphragm moves down, rib cage moves out. So what happened? Diaphragm moves down, rib cage moves out. So the volume of respiratory cavity is increased. Volume of the respiratory cavity is increased. So there is a formula. There is a formula. Pressure into volume is equal to constant. PV is equal to constant. So when volume is increased, pressure is reduced. Now the pressure inside the lungs is low, so the air is drawn in. So air moves from high pressure to low pressure area. So when diaphragm moves down, rib cage moves out, volume of this increases. So when volume increases, pressure is reduced. So air from outside, which is at lower pressure, moves in. Now when you breathe out, exhalation, what happens? Diaphragm moves out, rib cage moves in. So volume of the respiratory cavity is reduced. So when volume is reduced, pressure increases. Now pressure inside the lungs is more, outside is less. So the air is breathed, thrown out. That is there. Now coming to transport of gases. So basically there is transport of oxygen and CO2. So what happens? Oxygen combines with, first come to oxygen transport. Oxygen combines with hemoglobin to form oxyhemoglobin. Oxyhemoglobin. One hemoglobin can carry 4 molecules of oxygen. Now in hemoglobin you have presence of iron. Iron is present in ferrous form, that is Fe2 form. Hemoglobin contains what? Iron. So oxygen combines with hemoglobin to form oxyhemoglobin and that is transported. Okay. There are two interesting phenomena. One is carbon monoxide has 200 times more affinity for hemoglobin than oxygen. That is why carbon monoxide poisoning takes place. Then there is a thing called methemoglobinemia. Sometimes in our supply chain of food and water, nitrate comes. In our RBC, hemoglobin is present in Fe2 plus form, ferrous form. Now this nitrate will be absorbed in the stomach, it goes to blood, and here the Fe2 form is changed into Fe3 form, and such RBC have less oxygen carrying capacity. This condition is called methemoglobinemia. If it is a young child, the skin has a bluish tinge and this condition is called blue baby syndrome. So methemoglobinemia due to presence of nitrate, why Fe2 changes into Fe3 of the hemoglobin. The bluish change in case of young infant, this is called blue baby. Then when you go to high altitude, in geography you have different layers. So when you are rising up the mountain, so air density reduces and amount of oxygen is drastically reduced sometime at high altitude. And under that condition, the body comes under a condition called mountain sickness or acute mountain sickness. So when a person is going for trekking or hill, he should be very careful about acute mountain sickness. Sometimes it can even lead to death. Okay. So symptom, heavy breathing, nausea, dizziness, hallucination, headache, upset stomach. These are some of the symptoms. Just take a look over here. Mild acute mountain sickness, moderate, severe AMS, acute. So there can be water accumulation in the brain. There can be accumulation of fluid in the lungs, edema in the lungs and brain. So one must slowly move up the mountain. See what happens when you go slow up the mountain or when you relax at a certain altitude, then move up. When you go high up, oxygen is less, your body comes under oxygen deficiency. So in that case, kidney forms a hormone called erythropoietin, and this increases the formation of RBC. That is why people living at higher altitude have more amount of RBC as compared to people living in plain. So this is a normal acclimatization should take place. Okay. Now coming to carbon dioxide transport. Now if I come to, one is oxygen transport, another is carbon dioxide transport. So at tissue level, say in my hand at this place, CO2 is produced. This has come back to lungs. This is RBC. CO2 combines with water to form H2CO3. H2CO3 breaks into H plus plus HCO3 minus bicarbonate ion. Now HCO3 moves out into plasma. So most of CO2 is transported in form of bicarbonate ion in plasma. So bulk of the CO2 is transported in form of HCO3 minus ion in the plasma. Now in lungs what happens? In lungs HCO3 combines with H plus ion to form H2CO3 carbonic acid, and carbonic acid breaks into H2O plus CO2. That is what you breathe out. that you breathe out from the lungs. Okay. So this is the transport of CO2. There are certain disorders associated with respiration. One is asthma. So due to inflammation in bronchi or allergy, sometimes you have difficulty in breathing. So the breathing space is reduced. Then another is emphysema. It is a chronic disease. Sometimes the alveoli, deeper down the alveoli is affected, and if alveoli is affected, this condition is called emphysema. So smokers have greater chance of emphysema because the dust particle, because the smoke contains of the cigarette smoke also has particle or inhalation. Now these particles can cause many issues. So there is a concept called occupational respiratory disease. You are associated with a occupation where certain particles are inhaled. Now long term, when these are inhaled, it causes damage to the alveoli and other portion of lungs. So there are certain diseases occupation associated with occupation. One, you will find silicosis, inhalation of silica dust. In stone crushers, mining and etc., you have lot of this problem, silicosis. In stone cutting and chip making, all those things, you have silicosis. Another is anthracosis, coal dust inhalation. So these have to be reported. Coal dust inhalation. This is also called black lung disease. So in coal miners, this disease is very common. The coal mining industry has to take care. That is why specific gear or the your mask and other things should be provided for the safety of the employee. For the safety of the employee. Silicosis, anthracosis. Then beryllium, inhalation of beryllium compound is called berylliosis. In cotton and textile industry, the cotton fibers are very thin, they are inhaled. This is called white lung disease or bacinosis. Then asbestos particle, this inhalation is called asbestosis. So these are some of the respiratory disease associated with certain specific occupation. So we read about animals. Now botany is about plants. Now plant, you have different type of tissues. So tissues have two things. One is common function and common origin. Common function and common origin. So plant tissues are divided into two groups. One is meristematic, another is permanent. Plant tissues are divided into two groups, meristematic tissue and permanent tissues. Now meristematic tissue have the capacity of cell division. Meristematic tissue have the capacity of what? Cell division. Whereas permanent tissue do not have the capacity of cell division. So basically meristematic area are tissues are the area where you have growth in plant, and permanent tissue you will not have that much of cell division. So the meristematic tissue occur in the area of growth. Just take a look over here. The top portion of the plant, this is called apical meristem. Here apex, the shoot is growing up. Similarly, the root tip is going down. So these are called apical meristem. Apical meristem is found at root tip and shoot tip and the root tip. Okay, that is there. Then the girth of the plant will increase. The girth of the plant, a 50-year-old plant will have a greater girth, a 10-year or less, a 2-year less, still less. So the increase in the girth is due to lateral meristem. Now, so you have apical meristem found in root and stem, intercalary meristem, that is there, lateral meristem causing the growth of the girth of the plant. And while the apical meristem is growing, the plant grows up, some of the tissues can remain behind, and these are called axillary or lateral bud. So from here a branch or a flower can emerge from these axillary bud. As the apex goes up, some of the apical meristem tissues are left behind, and these form the axillary or the lateral bud. They are found in the axil of the leaves. Intercalary meristem is found between the mature tissues, and the animals like grazers, the cow, the goat, they browse it, they eat it. So if intercalary meristem is present there, they regenerate. So the grazing herbivores eat the top portion and the intercalary meristem regenerated. Lateral wall increasing the girth of the plant. The same diagram. Permanent tissues lack the capacity of cell division. Permanent tissue can be divided into two broad groups: simple permanent and complex permanent. Permanent tissue lack the capacity of cell division, divided into two broad groups: simple permanent and complex permanent. Simple permanent tissues have only one type of cell. Simple permanent tissue have only one type of cells, and complex tissue have few types of cell in them. So first come to simple permanent tissue. You have three main type of simple permanent tissue. One is parenchyma, second is collenchyma, and third is sclerenchyma. So the permanent tissue, simple permanent tissue are three types: parenchyma, collenchyma, and sclerenchyma. Parenchyma you will find primarily in leaves. These cell have, plant cell have cell wall, they have thin cell wall, they have thin cell wall, and they participate in photosynthesis. So in all the photosynthetic tissue you will find presence of parenchyma. Okay. So what is that? Their cell wall are thin, made up of cellulose, various function like photosynthesis, storage, secretion, that is there. Then collenchyma in patches. So here the corners are thickened. In parenchyma, this one, the cell wall are thin. In collenchyma, the cell wall become thick due to deposition of cellulose, hemicellulose, etc. So in collenchyma, here the corners are thicker. Why? More amount of cellulose and hemicellulose, pectin, etc. are there. They provide mechanical support to the growing part of plant, such as a young stem or the petiole of a leaf. The beginning portion of the leaf is called petiole. Okay. So parenchyma, collenchyma, and third is sclerenchyma. So permanent tissue three type, very thick wall. These are normally dead tissue. Here you have excess deposition of your cellulose, hemicellulose, and pectin. Sclerenchyma, long narrow cell, thick and lignin. Lignin is deposited, they are dead. Cellulose, hemicellulose, and lignin. Excess amount of lignin. So they are, most of them are dead, they are elongated. So they are found in fruit wall of nuts. For example, walnut and other, that is there. Pulp, hard substance in guava, pear, etc. that is there. Seed coat of legumes, the outer coat of pulse, moong, etc. are there, is what? Sclerenchyma. Leaf of tea is sclerenchyma. Now, coming to complex, permanent tissues are two types: simple permanent and complex permanent. Complex permanent can be divided into two broad groups: xylem and phloem. Xylem, the work of xylem is to carry water from roots to the top of the plant. That is the key thing to be remembered. Xylem function as a conduction tissue for water and mineral from root to the top of the plant. Whereas phloem, in xylem you have different component: trachea, vessel, xylem fiber, xylem parenchyma. You have four component in case of xylem. So here the question UPSC has asked is, transport of water from root to the top of the plant is done by what? Xylem. Now, to the top, the leaves receive the water, mineral, and other thing. They get carbon dioxide from the air and they do photosynthesis. After photosynthesis, food is made. Food has to be distributed to different part of the plant. So phloem will carry the food manufactured by the leaf to different part of plant. That is done by whom? That is done by phloem. Now phloem has four important components: phloem fiber, sieve tube, companion cell, and phloem parenchyma. So four are there. One I have highlighted is phloem fiber or bast fiber. It has phloem parenchyma, companion cell, etc. Phloem fiber you have to remember. So many of the fibers of commercial importance are basically phloem fiber or bast fiber. So basically they are what? Phloem fiber or bast fiber. So phloem carries food produced by the plant to different part of plant. So phloem fiber are commercially important. So fibers of jute, hemp, sun hemp, flax, they are basically phloem fiber or bast fiber. So to remove them, you have to cut the plant or you have to put them in water. In the water it decays. It decays in the water and then you have the formation of, then only they can be isolated. Okay. Then only they can be removed. This process is called rating. So jute and others, hemp, etc., you do rating. Flax, cannabis, from here also, one is bhang, cannabis also has flax fiber. So in many of the hilly states, they are using these fibers. Okay, one is for drug, another is for fiber. Cannabis, hemp fiber, bhang plant is there. So basically the tall variety of the hemp has fiber. Usually the stalk of tall cane-like male variety can be used in textile, insulation and others. Okay, so that is there. Nowadays, you have a plant called Himalayan nete. From here we also get fiber, bast fiber, Himalayan nete or Nilgiri nete. This is also being, we are focusing toward biological sources, natural fiber. Different Himalayan state government are trying to popularize the natural fiber. This nete, Himalayan nete, also the bast fiber is used. Now coming to the part of plants, plant have two part. One is vegetative part, another is the reproductive part. In vegetative part also, the part growing below the soil for absorption is your root and the portion above is the shoot, but some portion of shoot can be found inside the soil also. Now in root, just take a look over here, it is primary root, secondary root. Then take the case of shoot, here you have leaves, the branches arrive at the nodes. Okay, the distance between two nodes is called internode and here you have axillary bud. Then the stem is there, axillary bud is there, leaves are there. Then you have flower, bud, flower and fruit. So fruit and flowers are reproductive part of flower and root, stem, leaf and other thing are vegetative part of flower. These are the vegetative part of flower. Now plant with thin stem are called herb. Just take the A, B and C. Plant with thin stem are called herbs. They have short life. Plant with green and tender stem are herbs. Short life is there, few branches. Then shrub. Just above the ground, you will find large number of branches emerging just at the base of the stem. This is shrubs. Some plant develop branches near the base of stem. Stem is hard but not very thick. Bushes or shrubs. Then trees. Trees have tall stem. The branches arise in the tree above a certain limit. It is not from the ground. In case of shrubs, the branches arise from above the ground, whereas in case of tree, the branches arise at much higher level. So they are tall and have hard and thick stem. The branches at upper part, these are the trees. Then you have two more types, creepers and climbers. Creepers, they have weak stem. They do not grow up. So what they do? They grow on the ground. Such plants are called creepers. Weak stem, they cannot grow up and they are called creepers. So this is creeper, watermelon and many of the varieties are creeper. You will find growing near the sand at the river area. Then climbers. Climbers can twine around a support and can go up. Climbers. Okay. Now coming to modification in leaves. Leaves can be modified. Primary function of leaf is what? Photosynthesis. Primary function of leaf is photosynthesis, but they can also perform certain other function. Now just take a look. Tendrils. The leaf can be modified into tendrils. So these tendrils wound around a support and they help the plant go up. So this is tendrils. They are converted to tendril for climbing, as in pea, or into spines for defense purpose. For example, in case of cactus, the leaf is modified into spine. So this is a protective mechanism. So this is modification of leaf, here into tendril, here into spines. Now in some cases, the leaf will be short, so it falls down. Then the petiole of the leaf broadens and do photosynthesis. Another very interesting phenomenon is the leaf is modified into pitcher, or the, you have heard about insectivorous plant. One of the interesting insectivorous plant is pitcher plant. So in case of pitcher plant, this is the pitcher. Pitcher is a modified leaf. Pitcher is what? Pitcher is a modified leaf. So it is basically found in areas where soil is deficient in nitrogen. In India, you have only one indigenous pitcher plant called Nepenthes khasiana, found in Khasi Hills. So the pitcher has a lid. Here it has sticky substance. When the insect enters it, it is trapped. So now when the insect decays, the nitrogen is released. It does photosynthesis. It is not getting food by digesting insect. What it is getting from insect, it is getting nitrogen from insect. It does photosynthesis. So basically the pitcher is a modified leaf. The pitcher is a modified leaf. Very interesting modification you find in case of plant. Now coming to part of flowers. So this is a very common from example from NCERT. This is the rose flower. If you take the bud, the outer portion of the bud is green. This is sepal. All the sepals together are called calyx. Then you have petals. The brightly colored petals in different flower, you have different colored petals. The petals together are called corolla. The petals together are called corolla. So in flower, the sepal is the protective covering. Then you have petal, brightly colored insect pollinated plant. These act as interaction. Then the male portion of the androecium. Now androecium has a filament and an anther. The pollen grain are found where? They are found in the anther. So the male portion of the plant is stamen. Stamen has filament and anther. Anther contains what? Anther contains the pollen grain. Then you have gynoecium, or the female part, androecium, or the male part, and then you have gynoecium, or the female part. Gynoecium has three portions: ovary, style, and stigma. Ovary, style, and stigma. So basically, the ovules are found inside the ovary. The pollen grain comes to stigma, then a pollen tube will grow, and finally pollination or fertilization takes place inside the ovary. Pollination means transfer of pollen grain from anther to stigma. Anther to stigma. Ovary is in the enlarged basal part, then style, then stigma. After fertilization, the ovary changes into fruit. After fertilization, ovary changes into the fruit. So fruit is basically a modified ovary. Fruit. The fruit is characteristic of what? Flowering plant. After fertilization, the ovary develops into fruit. Fruit contains seed. Now just take a look. In the case of fruit, take the two cases. One is the mango, another is coconut. The outer layer of mango is epicarp. Then you have the fleshy portion, that is mesocarp. Then you have the hard structure, that is endocarp, and inside the endocarp is the seed. So here the portion that you are consuming is what? It is not the seed. You are consuming the pericarp, or the fleshy part, pericarp portion of the fruit. Whereas in case of coconut, epicarp is the outer portion, mesocarp is fibrous, and then endocarp is thick. Here you are consuming the endosperm portion. Here the endosperm, liquid endosperm, and then the solid sperm. So that is there. The ovules after fertilization develop into seed. Seed has a seed coat and embryo. Then in many plant produce certain chemical which we do not know why they are producing, and they are called secondary metabolite. For example, caffeine is there in tea and coffee. Why they are producing? What is their exact role in plant life? We do not know. That is why we call them as secondary metabolite. So different type of secondary metabolites are there. You can just take a look. Carotenoids, anthocyanin, morphine, codeine, opium we find from opiate plants, essential oil, many toxins are there. So what is their exact role in plant life? We do not know. That is why they are called secondary metabolites. These are found from plant. Now we discussed that plant has two portions, vegetative part, reproductive part. So if reproduction can be from reproductive part, that is normal. If vegetative part is taking part in giving birth to the next generation, that is called asexual or vegetative reproduction. So normal reproduction is through flowers, but if root, stem or leaves are participating in reproduction, that is called vegetative propagation. So vegetative propagation is example of asexual production in which new plant are produced from root, stem and leaves and buds, not from flower. Since the reproduction is through vegetative part of plant, it is known as vegetative propagation. So basically not from flower or bud, but from root, stem and leaves, then it is vegetative propagation. For example, sweet potato and dahlia, here the roots are giving rise to the next generation. So sweet potato is a modified root. If you put a sweet potato inside the soil, the next generation will come out. So basically root giving rise to the next generation. Then subaerial stems in vegetative propagation. Here one classical example is grass. You will find grass is put in patches and then it covers the entire area. Here this asexual production is called runner. So the stem arises at the nodes, new roots will come. These roots strike the ground and a new plantlet emerge. Slowly the entire area is covered. This you will find in case of grass. So this is example of a asexual production and these are runners. Subaerial, not fully on, it is on the ground. Then sucker. Sucker you will find in case of banana, bamboo, etc. So a lateral branch grows from the base of the stem. Then it grows laterally for some time inside the soil, where at the point where it comes out of the soil, new roots emerge. You will find this in chrysanthemum, bamboo, banana. This is example of sucker. So basically an underground portion of stem, it grows laterally, then comes out. Where it comes out, roots emerge, and this is the mother plant and this is the daughter plant. Take this. Then stolon, another place where the stem is giving rise to the next generation. So in case of runner, they were growing vertically on the soil. In case of stolon, it moves up into the air and then it touches the ground. Where it touches the ground, new plant develops. So runner, then stolon, then you have sucker. Now coming to these were subaerial stem modification. These were subaerial stem modification. Then coming to underground stem modification beneath the soil. Onion, potato, ginger, colocasia. These are example of underground stem modification. So potato and onion, these are example of modified stem. So if you leave potato, the eyes, you will find germination taking place. So these are what? Modified underground stem. So they store the nutrient. If you leave them, the eyes will come out and they will give rise to the new generation. That is why you store them in cold storage so that germination do not take place. Then leaf in plant modification. So this is found in bryophyllum. From the margin of the leaves, the buds develop. They develop roots also. When these roots touch the soil, these daughter plant move away and establish them as a new plant. So this is found in bryophyllum. The leaf giving rise to the next generation. Then you have, so these were natural. Now coming to man-made or artificial vegetative propagation, where human intervention is there. So here One classical case is stem cutting. This you have seen very common. In case of rose, China rose, or in case of sugarcane, you cut a portion of the plant and you put it in moist soil. You put it in moist soil. After some time, roots emerge from the lower portion of the stem and shoot from the upper portion. So this is your stem cutting. Just take a look. This is also there in your sugarcane. So not the entire, the long one is cut into short portion and you place them in soil. That is how the root and the shoot emerge from there. So this is your artificial, one classical case of artificial vegetative propagation is stem cutting. This you will find is rose, China rose and sugarcane. Then layering. In case of layering, the lower portion of the branches are put beneath the soil and then they are tugged onto it. So after some time what happens? Roots emerge from it. So layering, the lower branches are put inside the soil, covered with certain heavy object or with certain other things. So after some time roots emerge from the portion of stem which is beneath the soil, and this is the mother plant, the new plant. This is called layering. You will find this in grapes and other areas. This is a grape. So this is a layering. So this is your vegetative propagation. Then coming to some of the adaptation in plant and plant growth hormones. So plant growth hormones, the first one is auxin. Like in human, you have also in plant, you have hormones. So first hormone is auxin. So these are found at the tip of the stem and at the tip of the root. So it promotes cell division. Auxin causing cell division. So if the condition is growth, it will promote cell division. It enhances formation of root in stem cutting. Nowadays, many hormones are applied. You cut a portion of the branch, you dip in dilute auxin, you put it into soil, the roots emerge quickly. It also help in parthenocarpy, that is formation of seedless fruit. Nowadays, you have many variety of seedless fruit coming into picture. Then pre-harvest fruit drop. Achha. Nowadays, uh, dilute hormone spray is done on plant. For example, if flowering is less, if dilute auxin hormone is sprayed, flowering will increase. Whether it is mango or other plant, you use this hormone to enhance flowering. Then one synthetic auxin is 2,4-D. This has been used as weedicide. UPSC has asked in all exam. 2,4-D, 2,4-diphenoxy acid is a synthetic auxin used as a weedicide to kill the weeds. Now, this auxin is found at the top of the plant. So, what happens? The lateral branches growth are suppressed. The plant will grow vertically. So, you will find in the gardener what they do. Sometimes they cut the upper portion of the stem. When you cut it, the auxin is removed. So, what happens? The lateral buds emerge. So, it gives you a bushy phase. The lateral branches will come. So, bush-like appearance of the plant will come up. So, when you cut up the top portion, the lateral branches will grow. The growing apical bud inhibits growth of lateral buds. This phenomenon is called apical dominance. So, you cut the shoot tip, the lateral branches emerge. Then another hormone is gibberellin. So, if condition is good, see, if condition is good, the leaf will elongate, the stem will elongate, and the seed dormancy will be broken. So, you have your gram, moong, etc. in dry condition, it is not germinating. So, germination happens when favorable condition is there. Either water is there in the soil, or if you have put the seed in, you have soaked the seed. Breaks dormancy in certain crop like potato. Then it is more effective in inducing parthenocarpy, that is seedless fruit. So, gibberellin, auxin also causes parthenocarpy, that is seedless fruit, and gibberellin also causes. Gibberellin promotes germination and flowering. It means if condition is good, germination will take place, flowering will take place. For example, in March, April, condition is dry, but in June, July, moisture is there, water is there, temperature is favorable. What happens? Germination and flowering happens. So when you spray certain crop based hormone, the length of the stem will increase. It delays senescence. Senescence is the process of aging. So gibberellin will delay senescence, the leaf will not fall. Then you have another hormone, cytokinin. So this causes growth of lateral buds. Lateral buds were suppressed by auxin. So when you cut the top portion, the lateral buds emerge. This also delays aging. Then one hormone is ethylene. Ethylene hormone causes ripening. Natural ripening of the fruit is caused by this hormone, ethylene. Naturally ripe fruit will be juicy, whereas artificial ripened fruit is by carbide, that will not be juicy. Some portion would be hard. And ethylene is a gaseous hormone. Some fruit you will find the shopkeeper keep wrapped in paper or sometime in closed spaces. Okay. So natural high ripening is due to this ethylene, and this is produced in tissues which are undergoing aging or senescence. Then there is another hormone called abscisic acid. This causes abscission of fall of leaves. You will find in March, February, March, the leaf fall is there. So leaf fall is basically a mechanism to prevent loss of water or moisture. So during March, April period, after February, mid-February, the moisture content is reduced, and in India you will find in March lot of the leaf fall is there. So this is called abscission. So leaf fall is under the control of this hormone called abscisic acid. So if moreover leaf fall is there, it makes the seed dormant. If condition is adverse, the seed will not germinate. Then there is a synthetic thing called Agent Orange. This was used for removing leaf. This was used on large scale in Vietnam War as a defoliant. It is not a hormone. This chemical was spread over large area in the Mekong Delta in Vietnam. Still, you will find the effect is there. It causes cancer and other thing in. Then coming to ecological adaptation in plants. Each plant has to adapt according to the condition where it lives. First come to plant found in water, that is hydrophytes. So water is all around. If water is all around, what happens? The root have to do less work. So the size of the root is less. Stem thin and delicate. Why? To tolerate the flow of water. Leaves are either thin, linear or ribbon-like. Why? If it is not thin, linear, ribbon-like, the leaf can be torn away. Some plant have floating mechanism. This helps the plant to float. So some of the plant will have floating mechanism. So that is in hydrophyte. Plants found in dry condition are called what? Xerophyte. So xerophyte has to tide over what? Water deficiency. So since water is less, the root have to go deep and extensive. On surface, you have waxy coating on the leaves. So what happens? This prevents loss of water. Leaves are generally small, or leaves may be converted into spine. Leaves may be converted into spine. There is a tree, xerophytic adaptation. This is the khejri tree, that is the state tree of Rajasthan. The roots of khejri tree can go 30 to 40 meter deep. So this is a xerophytic adaptation. This is the state tree of Rajasthan. The tree might be 10-15 meter tall, but the root system can go 30-40 meter deep. Then come to a special type of vegetation called mangrove. This is found in coastal area. In coastal area, you have small evergreen tree. You will find this in Andaman Nicobar. You will find this in Sundarban, along the delta area and other. So these are evergreen tree. Water is all there. What all adaptation are there? See, since sea water is there all around, the soil is soaked. So therefore, they have a presence of a thing called breathing roots or pneumatophores. These root-like structure come out of the soil, and they help in breathing they help the roots in breathing. In case of agriculture, we first do tilling of the land. Why? To make the soil loose. Here these roots are coming out, and these are called breathing roots or pneumatophores. Presence of breathing root or pneumatophores. These are the breathing roots. These structure coming out of the soil. So when high tide is there, this will be covered, but the breathing roots will be peeping out. So there many animals lay their egg. Then second type of unique root is tilt root. This also you will find in case of mangrove, and that is why the mangrove tree will not fall down. If it is a normal tree, it will fall down. Mangrove roots. This is the stilt root of a mangrove plant. So it is there in the water, living happily. Second is seeds germinate while they are attached to the parent plant. This is unique. This is called vivipary. In mango tree, you will not find the mango germinating, whereas in case of mangrove tree, the seeds germinate when they are attached to the mother plant. This condition is called vivipary. So after germination, it drops and establishes as a new plant. These are some of the adaptation in the case of plants.